I would like to start the program. Please be seated.
I don't know. This is something that Derek really wants to do. And it is formal because it's Chapel Hill.
I would like to start the program. Please be seated. Good morning, ladies and gentlemen, and welcome to the 10th anniversary Annual Tech Roundtable. We know how busy your schedules are and appreciate you joining us. Today's event is part of the Technology Roundtable series by Nagoya University and its US Tech Transfer Office called NU Tech, or more formally, the Technology Partnership of Nagoya University. We are so happy to celebrate the 10th anniversary with you here today. My name is Shun Sekimoto, as the moderator of this event. Our 10th annual roundtable showcases cutting-edge biotechnology from Nagoya University, NC State University, and UNC Chapel Hill. We are very grateful for this partnership with two elite universities. Events like this one are possible only with the support of our corporate sponsors. Our sponsors are Jenkins, Wilson, Taylor, and Hunt as gold sponsors, and BASF, Meyer Burger, and Okaya USA Inc. as silver sponsors. Thank you to each of these outstanding businesses for your support. The Japan Science and Technology Agency, North Carolina Biotechnology Center, and Economic Development Partnership of North Carolina have cooperated with us to make today's event possible. We also appreciate JSP, MCPC, and EDPMC. The 5 technologies to be presented today are only a small sampling of the innovative work we are doing in the biotech field. We hope that today's roundtable will strengthen these critical networks and relationships between university researchers and business leaders. At this 10th anniversary event, in addition to technology presentations, we will have a panel discussion on how we can promote industry and academia collaboration and regional economic development. At this time, I would like to invite Dr. Akihiro Saso to offer opening remarks. Dr. Saso is the Vice President for Academic Research and Industry Academia Government Collaboration at Nagoya University. He visited RTP all the way from Nagoya City, Japan for this event. Ladies and gentlemen, please join me to welcome Dr. Saso.
Good morning,
Good morning.
distinguished guests and ladies and gentlemen. It is my great honor and pleasure for today to have AgriBio Roundtable jointly hosted by Nagoya University and its U.S. Technology Transfer Office, NU Tech. Let me thank everyone for attending this conference in spite of your busy morning. The NU Tech Roundtable series has been sponsored by Research Triangle Park Community, RTP. This is the 10th anniversary conference. On behalf of Nagoya University, I'd like to thank you for your continuous support. This year, we again focus on agri-biotechnology, which is, I believe, one of the most promising and strongest field in RTP. Today's event is held in cooperation with our partner, North Carolina State University, University of North Carolina, Chapel Hill, North Carolina Biotechnology Center, and Economic Development Partnership of North Carolina, and Japan Science and Technology Agency, and the general support by our sponsors, already introduced. We are honored to have a keynote speaker, Dr. Tom Adams from Pairwise. It is followed by 5 presentations, both from US and Japan side, about cutting-edge technology in this field. We also have guests from Japanese universities including GIF University, University of Education, and IT University of the Art. Thank you for coming all the way from Japan. I hope this roundtable will strengthen our network together with us. Nagano University is a home to 6 Nobel Prize laureates, and in enhancing its research activities in order to bring innovations. Establishing a new take in RTP is our strongest commitment to academia and industries in North Carolina and United States. Nagoya University, in Nagoya University, Institute of Transformative Biomolecules, ITBM, is active in basic research in agri-biochemical science. And 2 speakers comes from the Institute this morning. And as an application counterpart, we have established the Institute for Nanobiosystems in the Research Institute of Innovation for Future Societies. Moreover, from April next year, Nagoya University and GIF University, which has good schools of agriculture, start to merge in a budgetary sense first. This is a good news for us to boost research activities in this field. So finally, I'd like to thank our partners and sponsors and audiences for making this roundtable possible. I wish you a stimulating and productive conference. Thank you very much.
Thank you for your remarks, Dr. Sasso. Today, we are very fortunate to have with us Dr. Tom Adams, CEO of Pairwise, as our keynote speaker. By its strong and unique intellectual property position, Pairwise is a leader in the gene editing technology to create new crops and to improve existing ones through various research and partnerships. Dr. Adams kindly share Pairwise technologies and businesses with us today. Please join me to welcome Dr. Adams.
Thanks very much. I appreciate the opportunity to come and talk a little bit about Pairwise and gene editing and some of the things that are going on in this space. So I think for the talk today, I want to talk just a little bit about Pairwise. We're a pretty new company. I'll tell you a little bit about where we come from and what our goals and mission are. Then talk about some of the tools and opportunities that are arising in gene editing and how they matter. And then some of the things that we think that gene editing can do to really help improve crops and really, in many ways, improve food. So one of the things, when you start up a company, it's nice to have a purpose and a mission. And one of the things that we really realize is that it's a lot easier when you go into the grocery store to turn to the potato chip aisle sometimes when you want a snack than it is to go to the produce aisle. And we're not, all of you are not alone. Most people in America and many people in the world make that choice. And really, I think it comes down to maybe 20% of people in North America eat the right amounts of fruits, the recommended amounts of fruits and vegetables. So, we'll talk a little bit about why that is. But what we've really set out to do is try to take on this problem of, I guess 2 billion people in the world that have diet-related diseases. There's another 2 billion people who don't get enough food. So both of those problems are really important and really addressable. So we founded Pairwise really in April of 2018. Operationally founded. It was incorporated a little before that, but it was founded based on setting up a Series A from Deerfield and the Bayer Leaps group. It was Monsanto at the time that funded the startup, and then we did a collaboration on row crops, corn, soybean, wheat, canola, and cotton with Bayer Crop Science. And so really our strategy was the idea that we're going to collaborate in row crops where there are big companies with big interests and big acres, and then really use our resources to drive these what we'll call consumer crops. My co-founder operationally is Haven Baker, who's the chief business officer. He came out of Simplot and was responsible for developing the GM potatoes that are actually on the market today. So his background is both in biotech but also in business. So after a lot of searching around, we decided that RTP was a great place to put the company, and really have great facilities here. So those of you who aren't from the area, we have just a mile or two away from here, we have a greenhouse and growth facility that we're able to inherit from what's now Bayer, where we have 24,000 square feet of greenhouse, which for a startup company really gives us a lot of flexibility on things that we want to do, as well as a suite of growth chambers that can allow us to do a lot of experiments in them. And then a couple of months ago, we moved to where is now our core headquarters, which is in downtown Durham in the Golden Belt Complex. So about a 10-minute drive from here, 10 or 15 minutes, depending on traffic and whether there's been an accident on 147 or not. So we have really two nice facilities, and we now have about 90 employees. We have a really great opportunity with technology. We have three of our co-founders are the academics are on the right, David Liu from Harvard, Keith Young from Mass General, and Fong Zhang from the Broad Institute. And the three of these guys are kind of the East Coast leaders in technology around gene editing and improvements on gene editing. I'll talk a little bit about some of the things that are happening with them, but David is the inventor of what's called base editing. I'll talk about why that matters, but Pairwise has exclusive rights to base editing out of Harvard. Keith is, one of the shortcomings of CRISPR enzymes is, although the RNA guides, there's also a part of the protein, and that limits the sites that you can get to, and Keith has been really important in increasing the number of sites that you can move the enzyme to, so it gives a lot more access to the genome. And then Fong is responsible for really core base technology. In addition, we've got a bunch of IP from Bayer Crop Sciences that we can use broadly, which has been really helpful. So really, we have the capabilities to go all the way from base editing, automation, screening, transformation strategies, et cetera. So really a well set up base, and in 18 months since being founded, we have a really strong organization to work on that. So I wanted to talk just a little bit about gene editing and the technology. And one of the things we really got excited about gene editing a few years ago when the various discoverers showed that these bacterial enzymes could be guided at a particular site in the genome and create a cut. And that cut typically gets repaired by what's called non-homologous end joining. And that's a very imperfect process. It involves a lot of complex enzymes and really isn't editing, it's more like deleting, right? So you can create knockouts of genes at a pretty high efficiency, and that's been pretty effective when you want to knock out a gene. The problem is evolution isn't a series of loss of genes, right? The improvements that occur through evolution tend to be changes in activities and improvements to an activity. So differences in expression levels, differences in enzymatic activities that really drive the changes that we appreciate in nature that create all the diversity that we see. That's a really inefficient process. So what the initial gene editing approach is, people have really pushed on what they call homology-directed repair. So that's where you take a template, you cut the DNA, and you take a template, and you hope that template will direct repair of the cut. It's more like 1 in 10 to the 4 cells that it'll occur in. So really, with any plant transformation processes, incredibly inefficient. You can brute force your way to the changes you might want to make, but it's not very efficient. Base editing is a derivative of gene editing, or the original gene editing concept. And what it does is it inactivates the cutting piece. So you can take the same CRISPR enzyme, make it so it still homes in on a specific spot of the genome, but it doesn't cut. And instead of cutting, it has a new, it's been fused to another enzyme that modifies a base chemically. So it will modify a C, so it looks like a uridine, and then the uridine gets treated like a T instead of a C. You can also do this with an A and a G. And David Liu invented this technology. It's a really cool technology, and quite efficient, really just efficient as cutting. So if you want to make certain changes, it's very efficient at making those changes. And when you couple that with you couple that with improvements in the homing part of the CRISPR enzyme, you get to where you can almost move to on-demand editing of various sites. So I think what you really want to get to is I'd like to change any base to any base, and base editing gets you about halfway there. So just in October, so that's just last month, David Liu sort of one-upped his initial invention and came up with this new thing that he calls prime editing. And prime editing really gets you exactly where you want to be. So I think we haven't seen anything published yet on this in plants, but I think we'll see fairly soon. I know we'll see fairly soon things that are coming forward in plants. And it's a very efficient way to change any base to any base. So what he figured out was, CRISPR as an enzyme has a protein component and an RNA component. The RNA is how you direct the address for CRISPR to go to a particular site. So you can match the sequence in the DNA and the RNA and it'll find that. What David did was extend the RNA with a template. So I described that templated editing so that the RNA guide is longer, it has a template. And then he inactivated the cutting component again and fused, rather than to the enzyme I described before, now to a reverse transcriptase. So it'll actually turn that RNA into a DNA and it'll template that change that you put in the RNA right into it. So that's a sort of a little bit scientific explanation of what goes on, but the real key is that it now makes it so at about 30% efficiency, you can change any base to any base. It also takes away a lot of the limitations with the protein component because it's a much larger window you can make these changes over. So in humans, they've gone through and analyzed the ability with this technology as it stands today to repair every genetic mutation that's known. And there's a huge number of diseases that are known to be caused by genetic mutations. And using prime editing, theoretically, you could fix 95% of those diseases. So it's a really, really powerful tool that can really allow us to make essentially any of the changes that we want to make. So I think we're about to enter into the stage of real editing that you really can rewrite a gene to look the way you want it to. So with that tool in mind, and thinking back to our mission, what we really have set out with a couple of goals for Pairwise. One, we believe being product focused matters. So you can easily go down paths that with science that you do some really cool science, but it doesn't really have a product outcome. And then, gene editing and modifying technology in agriculture isn't always the most popular thing. So we have set out with the idea that we'd really like to do things that help to drive a social license for gene editing so it can be used pretty broadly. So as we've want to define what a social license means, what we've defined that is that consumers like the product enough that they don't really care about the technology. And I think you can think of lots of things where people really don't ask how you made it. Some in food. We all are carrying, I see a few people with their phone out. I don't really care when people tell me that the phone might cause brain cancer. I don't even look into whether that's true or not because I'm not going to give my phone up, right? I need my phone and that's just the way it is. So we look at the different business models that we could play in agriculture, and in the major row crops, people tend to focus on the farmer. Farmers love GMO products. Consumers didn't like them, even though consumers don't interact with them directly. They haven't felt the benefits in a direct way. Farmers adopted them incredibly quickly because they were pretty valuable to them. Then there's the ingredient space, and various companies are trying to be in the ingredient space. There your customer is really a food processor. You can talk about consumers, but the reality is it's a little bit substitutable in that if I make a better oil, you can find better oils in other plants and things that are already naturally there, so the uniqueness is hard. So what we've really focused on is things that consumers interact with directly, and the idea that you can make changes there. And I'll talk about some of the things that sort of support this idea. So we've focused on the fresh fruit, berries, and fresh vegetable markets where the consumer and the retailer are. And we're trying to look for traits where there's a demonstration in an analog where people have shown a willingness to both adopt the technology and support it by how they pay. So just to go back to this point that you know, the majority of Americans and really people in the world don't consume the recommended amounts of fruits and vegetables. And that hasn't changed in 50 years. So when you start thinking about why it hasn't changed, then you can look for the things that have driven consumption. And the willingness to pay and consumption are both linked to convenience. So you can identify several places where convenience has driven consumption, the availability of things, and of course taste plays into that. So, you know, examples are seedlessness. So most things that are seedless grapes, seedless watermelon, seedless mandarins, people are willing to pay 40 to 50% more for those, and they consume a lot more of them. I'll show you some examples of that. Small snack size. So snackability is a real driver in consumption these days, that people are, there's a lot of growth in the snack category, and I saw a statistic the other day that I think 60% of people would rather just snack, which seems kind of odd to me. I like to sit down for a meal, but snacking is getting more and more popular. Sweetness, colors, and off-season can be really important. So just to get to the examples, go back to 1986, so people who are a little younger think that we always ate these little baby carrots, but reality is carrots used to look like these long orange things with green stuff on top, and you'd slice them up. And the mid-'80s, there's some guys who figured out who misshapen carrots were being fed to cows. And these guys had the idea that let's take those and we'll stick them in a rock grinder and grind them down to these little things and see if people buy them. Well, people did buy them, and within 10 years there was a doubling of the amount of consumption of fresh carrots, and the price was significantly higher, I think 5x the price. So people were willing to both to pay for that processing piece, but also consumed a lot more carrots, or at least bought them. I don't know how many get put in a little plastic bag for your kid's lunch and they might not get eaten, but they do get consumed in one way or another. I think that's an example of convenience. It's not clearly a big flavor driver or anything, but people feel good that they're getting their kids some vegetable. Year-round availability is another really interesting thing. So if you take the blueberry case, and again, some of you might not realize that blueberries weren't always available year-round. They were really only available maybe a month or so during the summertime. Now you can get blueberries any day, and there's some variation in quality. That actually seems to be more genetic than it is because of shipping or storing. It's differences in the variety, so I think there's still some opportunity to improve this, but that was about 60 years of breeding, where people, a lot from University of Florida, but then other universities took on breeding northern varieties of blueberries with some southern varieties of berries and sort of changed the chill time. So they changed the geographies blueberries can be grown in, selected for some better structure. Now, you couple that with shipping, blueberries have a pretty good shelf life. So now a lot of them are, right now you're getting them from South America when you're buying blueberries every day. But those can be shipped for 60 days. Part of that's through breeding and selection lines that can be stored. Bottom line though is in about 2007, 2008, they became available year round, and the consumption of blueberries has gone through the roof in the last 10 years. So making them available year round drove a lot of consumption. And another example that brings in both the availability and the convenience is the easy peel mandarin, and these have become a staple, and I think almost probably 60% of kids' lunches, right? They're so easy to put into the lunch. They're a nice little size, and they're not messy like a navel orange when trying to peel it. I think navel oranges are better, but the reality is it's a lot easier to take a couple of cuties for your lunch or halos than it is to take a navel orange. And they've now become, and really just since 2013, they've become 2/3 of the citrus market. So total switch, and citrus was a dying space. There was less and less citrus being consumed, and mandarins have essentially caused that to be recovered. So I think there's a lot of really strong examples of how doing things that make it more convenient and easy to eat things drives consumption. So what we're looking at is a couple of things, is taking some things that are pretty healthy and driving their convenience. So One of our concepts, we've got cherries here, is to make a pitless cherry. Trees take a while to work on, so it's not going to be our first thing that we launch. But the other is actually an example, I think that's a black raspberry, but blackberries and black raspberries, and these are very healthy berries, and we think we can make those more popular and better tasting varieties available. And we're also looking at some things with vegetables. So a number of product concepts to drive, again, availability year-round, convenience, and snackability. So the berries are actually pretty interesting. The rubus genus is pretty interesting. There's like 740 different varieties of rubus. We only eat 2 of them, right? Blackberries and red raspberries, but there's a whole lot of other berries. So we've, and if you look across the spectrum, most berries are like weeds, brambles, and you can see them out in the wild. And then there's things that have been, raspberries have actually been almost completely grown now in these hoop houses. So they're grown indoors and they're grown in pots. So I hadn't really realized this until we started to learn about the industry, but all of these things are being taken out of the soil and grown in pots because of the lack of the nematicides, the fumigants being taken off the market, so the best way to grow these things. So they're really becoming sort of annual crops rather than things that you plant for 10 years and then take them out and replace them. So it's become a very different industry. Blackberries, they sort of tie up in trellises, but there's a lot of opportunity still to move them over to the right. And then, I think the big question is how indoor farming is going to affect this. But in the meantime, you're almost indoors with raspberries, and they're available year-round. Now, are they the best tasting ones available year-round? I think there's still some opportunity there. So we've been working on the berry space and trying to, basically our concept is we can use gene editing as a way to domesticate some of these berries and get more variety and different berries available. This just shows that we've developed a transformation system for cane berries, and we are, which is sort of one of the keys of doing gene editing that we don't think about is that you have to actually be able to regenerate a plant in order to edit it. So it's not, I can just say I want to do a cherry and I can do a cherry. We have to develop a system. We're just doing an inventory of the staff that we've hired in transformation, and they've actually got a history of transforming 65 different plants. So there's a fair number of plants that have been transformed, and we feel pretty good about being able to solve these problems. But in a bigger sense, what we also want to really understand how to improve them and which varieties to improve. And we put together a public-private collaboration with basically every plant breeder in North America, or every berry breeder in North America. So NC State happens to have a pretty good cane berry breeding program. Arkansas is probably the biggest one. The USDA has a facility at Oregon State that basically cultivates and maintains all of those 740 varieties. Plant Sciences is a company that provides germplasm that sort of competes with Driscoll's in that space, and BC Blueberries is British Columbia, and then Cornell is part of this. So these are basically all the berry breeders. We've got all the varieties, some of the things that are commercial and some that are wild, and we've sequenced several hundred of these, a few hundred of them already. So we've done past the 30-40x genome coverage, doing a few of them at a really high level. And then the academics are going to work with us. We've got a phenotyping trial set up. Both they're running some phenotyping trials, and then we're running a trial in California with Plant Sciences, where all of these different berries are being grown under hoop houses in pots. So you can see all of these phenotypes and associate them with the genetic variation. So things like thornless, for instance, which is one of the reasons we have, I don't know how many people buy blackberries, but if you've ever eaten a good blackberry, you know the ones you buy are not really good blackberries. And that's partly because the thornless varieties are not that good. And it takes a breeder a long time to move thornlessness into the blackberries that taste good. Thornless is actually a pretty simple trait. You can see the segregation of thornless, and I think it showed, so this is just showing you can start to divide the phylogenies, and I think this is an example here. You can see pictures of you can see these, but this is a thornless variety here, this one has some little bumps, and this one has bigger thorns, here's some nasty thorns. So you can see grouping of these varieties into these different categories, and begin to make associations between genes and which are the mutations that you need to make using tools like base editing to do that. So lots and lots of opportunity in this space to make berries, which are one of the faster growing areas in the produce aisle, more approachable. So I think I mentioned this about the big phenotyping effort. This is the site where we're doing this with the academic group, and all be growing in these kind of hoop houses. A little bit hard to do this where some of them are essentially weeds, so a little bit tricky, but I think a lot of exciting data should come out of this. So with that, I think I've given you a little bit of a feeling for where gene editing can have an impact, not just on agriculture, but on food, and we're excited to try to make a difference in people's diets. Happy to take any questions.
Are a lot of the traits you're going after, like seedless and things like this, amenable to using CRISPR or gene editing to solve?
Yeah, I mean, I think, so the question was basically, are the traits that we're interested in things that are amenable to gene editing? So one of the things that, probably the last 30 years of plant science has really done quite a lot to describe a lot of pathways that affect some really important things. So Steve mentioned seedlessness. So one of the things that we're very interested in is removing the seeds in blackberries, which I actually never noticed that much, but we did a consumer survey, there about 90% of people said they'd pay more for blackberries without seeds in them. So it seems like that does limit some people from eating them. So seedless, well understood, for instance, what makes a seedless grape, these types of approaches, and those are editable traits. So it's really just altered function in certain genes. Flowering time is really important to be able to extend seasons, and changing that. A lot is understood about flowering and all the environmental integration for flowering that occurs. So, the idea took 60 years of breeding to make blueberries have the range that they have today. I think it's not like you can just program it exactly, but you know exactly where to hone in and start making mutations that will repeat in cherries what you have in blueberries. So year-round cherries would be a pretty valuable product.
I'm so wondering, isn't that aren't these traits or modifications you could also achieve by breeding, and it just hasn't been done because the breeders didn't focus on it or whatever, they're not smart enough. I thought, okay, in medicine you are really fixing mutations or diseases and curing people. That's a big deal, of course. I wonder, what is the big deal for agriculture? Is it like creating gene-edited, whatever, purple tomatoes with rich on some anthocyanins, something which you could breed maybe?
So there are maybe two things to add. So I think that's a play of gene editing, that there are some new alleles that maybe don't exist in nature that you could breed. The way that it's being approached in public and regulatory now, there is a lot more acceptance of doing things that could be achieved by breeding, but you can do them a lot faster. So I think the first set of traits ideally are things that are easy for people to get their head around, which are things that probably you could achieve by breeding. But as I mentioned, the blueberry example is 60 years of breeding. So a cherry, sour cherries, the ones you make cherry pies with, that tree was bred 400 years ago, before the French Revolution. It's from France, it's called Montmorency. That tree is, and it's the same tree, it's clones of that exact same tree. So a lot of these species that we eat, especially in the fruit space, are clonally reproduced, so there's very little breeding. And those clonal reproduction systems are great for gene editing, because now if I have variety, I can bring it into the clonal system and now add whether it's a disease resistance or a new trait. So I think there's an interrelationship between natural variation and then potentially it could move to new variation that would be, totally novel, but that is getting closer to where the public gets worried about pregnancies.
I'm afraid that. On to the next problem.
Thank you.
Thank you, Dr. Adams, for your interesting presentation. I understand pairwise will surely bring new crops and vegetables, but it is much faster through gene editing technology and other methods. Now, we will shift gears to our technology presentation, beginning with Dr. Masahiro Kanaoka. Dr. Kanaoka is developing researcher and lecturer at the Institute of Transformative Biomolecules at Nagoya University. He will talk about challenges to understand and manipulate fertilization. And ladies and gentlemen, please welcome Dr. Kanaoka.
Okay, good morning, everyone. First, I would like to thank for organizers to give me a chance to talk about my research here. So today, I would like to introduce research which is titled as Challenge to Understand and Manipulate Plant Fertilization. So as you already you are very familiar with this, so many food are from plant reproductive tissues. For example, the rice, what we eat are the endosperm of the rice, and corn also. And for example, the tomatoes, we eat the cattle of the fruit. So these are all of them are from the reproductive tissue of the plant. So to understand plant fertilization is also very important for sustainable and suitable food production. Okay, so I will briefly introduce the basic mechanism of the plant fertilization. First, the pollen grain from the male is attached to the female stigma, and then the pollen grain hydrate and pollen tube, this tubular unicellular cell is elongating inside the female tissue. And this pollen tube is, the growth is regulated by some signal from the female tissues. For example, some long-range signals may be secreted from the ovary of this part, the seed part, and the pollen tube is come close to ovule. Then after that, they have received another signal from the female gametophyte, the egg cell, and then it changes the direction to the final ball for fertilization. And this kind of signal interaction between male and female are not only important for the proper fertilization, but also the interaction works as a species barrier. So when we pollinate the pollen grain from the several different species to one species, in this case this is Argos thaliana as a model, some pollen grain may not germinate here, they still remain here. Some pollen grain can germinate and grow inside this tube. However, they may stop here due to the improper species recognition. Some pollen grain from some species can grow more deep inside and come close to ovule, but they cannot find the target and go wandering. So this kind of, to understand the molecular mechanism of male-female interaction is also important to understand the species barrier and to replicate the barrier. So my research purposes and my research interests are to understand molecular mechanism in plant reproduction, especially pollen tube guidance, and to understand reproduce the pollen tube attraction in vitro and break down the reproductive barriers. So today I would like to talk these three small topics. And first topic is anastomosis lure, which is the pollen tube attraction for species in a species-specific manner. So, as you know, the fertilization occurs deep inside the female tissue, so we cannot directly observe the plant fertilization because it's because the plant tissue is not transparent, you cannot see them. So to analyze and observe the fertilization, we make an in vitro fertilization system. So, here we dissect the female tissues and take them out, culture in the in vitro medium, and then do the assays. So by doing this kind of establishing an in vitro system, we can manipulate and precisely analyze the fertilization. So this is one example of the plant, what we found. So, this is an ovule, the female part of the plant called Torrenia, a kind of an ornament flower commonly found in garden. And this is the deep growing pollen tubes. So when we co-culture the pollen tube and ovules here, and the pollen tube try to reach to the female side to get inside here for fertilization. However, when we manipulate this one, you can see that the pollen tubes can chasing the female tissue like this. Chasing, and go. Yeah, so this really shows that something is secreted from the female tissue to attract pollen tubes, then attract the male. And the male is always chasing this part, so this part is the key to secrete the molecules, and the tip of the pollen tube is sensing the signals and precisely chasing the female. So now we know that the attractant, the pollen tube attractant molecule is a small peptide that we have published a couple of years ago. We named it Lua protein. The Lua is a very small protein with 6 system residues to make a disulfide, intermolecular disulfide bond. And this protein is secreted from the cells called synapse cell, which is adjacent to the egg cell to attract pollen tubes. So this is a pollen tube growing the medium. So usually they grow straight, but when we apply the pollen Lua protein to the mark asterisk, then the pollen tube changes its direction. So this shows that the protein still has the ability to change attract pollen tubes. And this story was first started from Torrenia, and now we know we have Lua asclepiae, Arabidopsis too. So we have 2 Luas in Torrenia and 1 family of Luas in Arabidopsis, but the sequences, the protein sequence, is totally diverse. So the only concept we have, there are only concept 16 residues asterisks marked here, but most of the other amino acids are not concerned. Although they have the same activity to attract pollen tubes, the sequence is totally different. So we are wondering which part is important for the function, meaning to attract pollen tubes. Which part is important and which part is not important, and which part is important for species-specific recognition. So we try to precisely analyze the protein function by making the chimeric protein between these 2 different Lua proteins. So again, the green bar shows the Lua from Arabidopsis, the blue bar shows the Lua from the Torrenia, T8. So we made a series of chimeric protein between these 2 proteins, like showing these color bars here. And the conclusion is that this part is the key to attract Arabidopsis pollen tubes. So for this concept, most of the part is the blue, which means the sequences are from Torrenia, and only this part is from Arabidopsis, but still can attract Arabidopsis pollen tubes. So which is this part is very important for pollen tube attraction in Arabidopsis. So now we narrow down this region again, and finally we found that only 3 amino acids is necessary and sufficient to change to target the Arabidopsis pollen tubes. So this concept has most of the part is Torrenia, and only we replace 3 amino acids from Torrenia to Arabidopsis, and that can attract Arabidopsis folensis like this. So Torrenia and Arabidopsis are totally different species. Phylogenetically, they are not related species. However, and the Lua sequence also different. However, by introducing some key amino acids, we can make this kind of, we can change the feature of the attractant. So it's like we can change the feature of the key to acid. And we also know that now we know the receptor of this Lua protein, and we have the crystal structure of receptor and the Lua proteins, and these three amino acids are facing to receptor side, and this is important for the receptor ligand recognition. Okay, the second topic is fabrication of the microfluidic device for analysis. So again, let's go again to the Torrenia. So this is the Torrenia ovary, which contains many ovules here, and this is a cut style right here, and the point tubes growing inside here, but you can see. But then after several hours later, the point tubes will grow outside from here, and you can see the point tubes growing on the medium, and they change its direction to the ovule side. So this ovules are the point tubes in this kind of long range. So to analyze, so we are interested in what kind of molecules attract the point tubes in this kind of long range. To analyze them, so we made this small chamber like a small pit using the microdust technique. So here we have a very narrow flow channel here to let point tubes grow here. And this channel is divided into two sides. So the point tubes has a choice to grow left side or right side. So if something is has ability to attract point tubes, the point tubes should go there. And if the compounds have no ability to attract point tubes, they don't go this side. So this is the hypothesis and make this kind of platform to analyze. So we put the ovules here. You can see the point tubes growing from top. All of them change to the left direction and to attract to the ovules like this. And the point tubes don't go to the blank side. So this tool is very easy and useful to analyze point tube growth direction. And by using this system, we found a new novel protein called HOVA, which can attract point tubes. So this is this protein is also cysteine-rich protein, but it's not related to the gluon. So this is a different family of cysteine-rich proteins that attract point tubes. And now we are doing the chemical screening to that they affect to identify what kind of molecules affect the point tube growth or its growth or attraction. So we have many thousands of chemical compounds in our institute, and we are trying to we are planning them to see the point tube behavior. So in this case, we put the ovule and some compounds simultaneously here. So this compound seems to prevent point tube growth to this side. So all the point tubes go straight or go the other side. So this kind of the microfluidic device platform is very useful for the screening for compounds. Okay, so lastly, I'd like to introduce the establishment of the in vitro point tube attraction assay in tomato. So the research I told here was done by using these model plant species, and we know that the sequence of the Lua point tube attractant was totally different from species to species. And in the monopod, for example, Siemens, researchers have reported that this protein is point chip attractant in the monopod. But this one protein doesn't share the sequence similarity or protein structure to the dipole dual proteins. So my question, I'm wondering how the attractant protein is different from species. What is the key feature for the point chip attractant? So I want to expand our knowledge. So I want to start a new project to identify point chip attractant from the other species. So I choose tomato because tomato is a very common plant and food product. And in the same family of Solanaceae, there are so many useful plant species such as potato, eggplant, tobacco, and so on. So by understanding the tomato, we will expand the knowledge to these Solanaceae family. So I will first try to establish the in vitro culture system. So the tomato is also used as a pollen tube research about pollen tube elongation or something. So we first we use the similar medium, already published medium, but we realized that this medium is not sufficient for the long-term analysis of the incubation of these tissues. The pollen tube can grow only for 1 hour or 2 hours, but it's, for our purpose, it's not enough. We want to keep them for 1 day or 2 days or more longer, possibly more longer. So we want to modify them, improve the growth condition. And first, we know that the protein called arabinogalactan protein, which has a very long sugar chain on the outside of the protein, is very abundantly produced in female tissue, such as ovule or placenta, where the pollen is growing. So we try to supply this AGP to the medium to improve the pollen condition. And we found that the commercially available AGPs, gram-arabic or amor, which this is our product from our institute, this kind of structure. This has a ability to elongate pollen tube more longer, and also by adding these compounds, the pollen tubes, the rupture of pollen tubes more or less decreased compared to normal medium. So these compounds help to keep the pollen tubes in better condition. And we also realized that the female part, the ovule, is not soon oxidized. You can see the brown color here. So by dissecting the female tissue, the ovule is soon oxidized and soon die. So we try to add some antioxidants such as vitamin C, and we also add the trehalose, the sugar, which may help to retain the cell membrane very strong. So, and then the browning of the ovule is reduced by adding these compounds. So now we have succeeded improved our growth culture condition for tomato ovules and pollen chicks. So this is the assay of the pollen chip attraction in tomato. So this is the tip of the pollen chip, and we place the ovule in front of the pollen chip, and then the pollen chip, 25 minutes later, the pollen chip changes the direction to the ovule side. So which means that this pollen chip is intact in vitro condition and is reacting to the ovules. And this, the bottom part shows that the pollen chip is growing more longer, and then they are entering inside the ovules for fertilization. So now we have succeeded in making this kind of pollen chip attraction and fertilization system in tomato in vitro. Okay, so So our future direction is to, of course, one is to understand the basic mechanism of the forensic guidance and fertilization, and the second aim is to apply this technique to the breeding. For example, in normal breeding, the breeders pollinate the pollen grain to the female pistil and wait for seed production. If they can get a seed, then it's okay. But in some cases, they cannot breed to the ovule. They don't have a sibling due to the incompatibility. But as I showed, that the in vitro system may break down this this problem. For example, in vitro condition, for example, if there is a problem about point elongation, then we can skip this stage in vitro condition. So we can use a more short style, and the point can easily reach to the ovule. Or if we can make a transgenic plant, we can introduce the attractant to the ovule of the ovule from different species, and then the foreign tube will easily be attracted. So this is one example of to show the performance of the in vitro culture system. So this is the female Saliana, and when we pollinate Capsula, the different Brassicaceae species, the pollen tube can grow like this. However, the pollen tube cannot target the ovules. They don't recognize the ovules, analogous ovules in vivo condition, so we can't get to see. However, when we dissect the pistil and take the ovules out to the culture medium, the capsule pollen tubes can come closer to the ovule, and then they can go inside the ovules in vitro condition. So the problem, so in this case, the problem was pollen tube recognition here, so we can bypass the barrier by directly placing the ovule inside in front of the pollen tubes in vitro condition. And this can be also applied for the crop species. For example, this is I use two different cultivar tomatoes. So the pistil and the pollen grain is from the microton, and the ovule is also the different cultivar tomatoes. So we can get in these heterogenic conditions. And I think we can apply this technique to the, for example, the self-incompatible plants. Or in some plants which has a self-incompatibility, we cannot get the homozygous seed because of the prevention in breeding. However, when we can, by using this kind of in vitro system, we can bypass the self-incompatibility reaction, and possibly we can get the homozygous seed by using this technique. Okay, so this is the end of my talk. Thank you so much, and I would like to have a comment on these questions. Thank you.
Thank you, Mr. Kanagawa. Let's turn to you in the audience now for questions. Is there any questions?
Do you know which cell in the ovule secrete? Do you know which cell the ovule secrete the attractants to the hormone?
Okay, in case of lure protein, it is secreted from the synaptic cells, which is located adjacent to the exon, this is S5 cells. For hormone protein, which is has a more longer attractant, this is secreted from the sporophytic tissue of the ovule, outside of the female.
Thank you again, Dr. Kanagawa. Our next technology presenter is Dr. Jin Lichten from NC State University. He is William E. Reynolds Distinguished Professor of Plant Pathology and Director of Emerging Plant Disease and Global Food Security Cluster. Today, Dr. Liseno will talk about her recent research on how we can tackle emerging plant disease outbreaks. Okay, ladies and gentlemen, please welcome Dr. Liseno.
Good morning. Thank you. I'm happy to be here today to talk to the group about some of the research we're doing. I lead the Emerging Plant Disease and Global Food Security Cluster at NC State. And I'm going to talk to you this morning about emerging plant diseases and how we're going to tackle the challenges of the future in managing these diseases. We know that the population by 2050 is going to exceed 10 billion on this earth, and we're going to have to figure out better ways to grow plants and grow food in a changing climate. Food production has to increase while we protect the ecosystem, reduce environmental impacts, improve water efficiency of crop growth, reduce energy costs, and then we have to deal with issues related to utilization and waste of food. Over 40% of the food that's produced is waste on the earth, and that's a problem. We can increase yields, but if we waste more than close to half the food, we have huge issues. And we all know, for those of you who live here in North Carolina, that we've had many climate-related disasters recently, including Hurricane Dorian that devastated the Bahamas and parts of the east coast of the U.S. We have an increase in these frequencies of storms and floods and hurricanes and extreme temperature events, and these are all leading to more disease outbreaks, and they're causing crop stress for growing plants. So by 2050, we have to increase food production by 60%, according to some statistics from the FAO. And this year, FAO has a big report out on food waste and how we're going to manage this food waste problem that I talked about. Plant diseases are a component of food waste because many of the storage rots that happen post-harvest happen because of plant diseases. So FAO published this diagram of the future challenges of food production, and one of the areas here that I circle is preventing transboundary and emerging agricultural pests and pathogens. We have outbreaks of pests and pathogens that are reducing food production in many areas of the world, including sub-Saharan Africa now, where they're dealing with a fall armyworm outbreak from a really devastating insect pest. So this is on the radar screen of all the development agencies, and also many scientists, such as myself in the cluster that I lead at NC State. This diagram is from a paper by Dan Berber at Exeter University showing that plant diseases and emerging pests and pathogens are on the increase. They did a big data mining study and showed that in North America, the slope of this curve is pretty steep, but we see increases in many other areas of the world. We have more reporting of diseases and pests in North America, so that's why there are larger numbers shown here, but in general, we've got emerging outbreaks happening globally. So why do diseases emerge? They emerge when we have an increase in geography or host range. A pathogen might shift to a different host. We might have change in pathogenesis or a newly evolved species, or even just a new race that causes problems. And then we have newly discovered pathogens and pests. So I'm going to give you a couple examples and then drill down on the work that I do on lake blight. Stem rust caused by Coccinia graminis is a serious problem on wheat globally, but in Uganda, a new strain was found called UG99 in this part of East Africa, in Uganda and Ethiopia. So what's happened is a lot of breeders are working now to try and deploy resistant varieties in the Middle East and in Pakistan and India, so you can prevent this outbreak from really destroying the green revolution that happened from breeding new wheat varieties with resistance to stem rust. And so this takes a lot of new technology, such as was just discussed this morning with some of the CRISPR-Cas9 work. And we have a faculty member in our department, Dave Marshall, that works with USDA on breeding disease resistance and trying to get ahead of this epidemic wave to prevent this outbreak from destroying food production in areas of the world where there's a great need for more food. And recently in Sicily, in 2016, a new race of the stem rust pathogen was found that probably migrated from North Africa, and this strain could break the resistance of most of the European wheat. And so this is a problem as well, so we're playing this arms game, trying to catch up with the new races of the pathogens as they emerge. In 2013 and 14, a new strain of the coffee rust pathogen hit Central America. This is what the disease looks like on coffee. And it occurred at higher altitudes where growers hadn't had to spray fungicides before, and we saw devastation of the coffee plantations. And essentially 400,000 workers lost their livelihood and many migrated. And in fact, there was a big article in the Washington Post this summer suggesting that much of the migration from Honduras and Guatemala could be related to lack of livelihoods in some of these areas because of coffee rust. So the cup of coffee that you're drinking this morning impacts livelihoods of people below our border. Another really important disease, Panama disease, tropical race 4. It's a new race of Fusarium, and it causes this devastating wilt on bananas, and it's recently been found in Colombia. We grow one cultivar of banana in Central America, Cavendish. It's clonally reproduced. It's susceptible to this race. So we have a pretty dire situation now if this lineage moves into Central America. And I've been working with Dole. We're going to come up with some technologies for detection so we can try and prevent the spread into the major banana growing areas. Most of the U.S. and Europe get their bananas from these plantations in Central America. The pathogen originated in Southeast Asia, but it moves in the banana corms and the planting material, and it's moved into Africa. It moves in soils. It's in Mozambique. They've had to quarantine. They can't fumigate. It's deep in the soil, so it stays for years. So it's going to require some pretty quick breeding of bananas. There are some transgenic bananas that are available, and people are working on gene editing bananas as well. So we have a crop that can be replanted in these areas. I work on the pathogen that caused the Irish famine, Phytophthora infestans. This is an Irish family in the 1840s uncovering their only food source, potatoes, that was destroyed by this pathogen. But this pathogen is not just of historical significance. It causes disease in Central America and globally, where growers have little access to the suite of fungicides that we have here in the U.S. So many times they don't spray, or they spray with the wrong compound, and then they have disease problems. So late blight is a re-emerging disease because it moves aerially. It also moves in potato tubers. It has a very rapid life cycle, so it can reproduce quickly. We have fungicide-resistant strains. We have the ability of the pathogen to move to different hosts. And the genome is, we sequence the genome, and it has a suite of effectors that allow this pathogen to overcome host R genes that are thrown at it. So it's a moving target. And then we have monoculture susceptible potatoes. There are some resistant potatoes being grown somewhere. It was mentioned this morning about Simpox potatoes with some resistance to late blight, but we have most potatoes globally that are planted are susceptible. So we're not deploying resistance where it needs to be. These are symptoms of the disease on potato leaves and tubers and on tomatoes and tomato fruit. In 2009, we had a major outbreak of late blight because the pathogen was distributed on tomato transplants that people brought into their home gardens. These transplants had lesions that were kind of hidden. Homeowners didn't recognize it. They planted these tomatoes in their home gardens, and it spread to tomato and potato fields, and we had a major meltdown of tomatoes, and it was reported in Science and the New York Times. And a group of us got together after this outbreak. My lab identified the strain as US22, and we actually were able to see when we did Google Ngram searches, this peak in searches for what's blighting my tomatoes. Every summer people try and figure out what's causing disease, but in that summer we had a huge peak, and we could see both tomato and late blight searches going on the internet. So the Google Ngram searches actually allowed us to see this epidemic emerging. Usually you'll have a pre-event, the movement of an infected tomato plant, and then you get detection, and then the epidemic may escalate, and the scientists and researchers get in usually down here after the disease is already spread, trying to figure out how to control it. So we want to be working in this space, in this pre-epidemic, pre-event space, and with some of the technologies we're developing. So we are run a USA blight disease surveillance system where we map disease occurrence. And we are also developing new diagnostics. We're genotyping lineages of the pathogen using 12-plex microsats. We have a fungicide decision support tool that was developed at Cornell. It's based on BlightCast, weather-based tool to tell growers if the weather is conducive when to spray. So we have a whole suite of technologies that we've developed to help in managing this disease. And we track lineages. That lineage that caused a big problem on tomatoes in 2009, this purple lineage, has pretty much disappeared. And we had this widespread displacement of that lineage by US 23. The lineage is sensitive to mefenoxam, which is a good thing, because previously we had these two other lineages, US 8 and 23, that were really resistant to mefenoxam. This is Ridamel is the common name for this fungicide. And now the lineages that are widespread in the US are sensitive to mefenoxam plus all these other fungicides. So the team tested the baseline sensitivity of all the lineages, and we have about one major lineage now. So we're pretty good on types of compounds that can be used to control the disease. And we're also developing detection technologies. We started out developing a LAMP assay for detection of the pathogen. It's based on cybergreen isothermal assay. It turns green if 3 tubes on the left are positive, these are negative, and then we can read these samples into a 3D printed reader on a tablet computer and then upload the data into a cloud source for recording outbreaks. We've also labeled the primers with indicator dyes so that we can run them through lateral flow devices, and you get 2 bands if it's positive, 1 band if it's negative. But the other thing that we're working on, I lead the Emerging Plant Disease and Global Food Security Cluster, and we hired Dr. Qishan Wei from UCLA. He's an assistant professor in chemical and biomolecular engineering. And we're developing sensors for detection of this pathogen and a number of other plant pathogens. Basically, when a pathogen approaches a plant, before you see symptoms, the plant starts emitting volatile compounds. And these volatile compounds, we can capture them using colorimetric sensors that are based on golden nanoparticles. And then we also have another kind of detection method we're working on. It's a microneedle patch. It's a small patch that can be pressed into the plant. If you've ever had a TB test, you know what a microneedle feels like. Basically, you press the microneedle into the plant material, and then the DNA can be washed off and you can amplify it within a matter of minutes. So we're moving both of these two technologies to the field. Basically, the first one, the microneedle patch assay, we can PCR from this, and we've recently found that we can genotype from the patch. We can sequence microsatellites, and we're doing Illumina sequencing from DNA from patch within 1 minute. So this technology could revolutionize genotyping, make it quicker and faster for companies to genotype. The other technology that we're working on, the volatile technology, we can distinguish infected from diseased plants. And the way this works, this is the patch. Let me show you the patch first. The needles go into the mesophyll past the epidermis. We extract, and we could, as I said, we could amplify DNA by PCR, but also by microsats, and now we're doing Illumina sequencing. This was published in ACS, American Chemical Society Nano, this summer. And then the volatile sensor, we detect pathogens using a colorimetric sensor. We've been able to sort out 3 different plant pathogens on tomato. That's our model system: Phytophthora infestans, early blight, Alternaria solani, and then Septoria lycophrosisi, which is another foliar pathogen. And basically, we inoculate the plants, wait 2 days, and then we remove a leaf, and we collect the headspace gas from around the leaf, because the leaf is responding to the pathogen, emitting volatiles. We've run mass spec, we've figured out what the volatile profiles look like, and we've developed these sensors so that they will detect the common volatiles that are emitted when these pathogens infect. And you see the different color changes, and those color changes allow us to use machine learning to identify the pathogen. We sorted out these 3 species using the sensors, and we're now working on a project where we're going to use these sensors for bacteria and for viruses and for other pathogens on tomatoes. And I'm also working on a project with Dole, because the TR4 race of the banana pathogen, Fusarium, produces volatiles in culture, and it also produces them in infected plants. And we want to help the companies be able to sort out race 4 from race 1. Race 1's been there for years and is not a problem, but race 4 is the killer race. So we're developing a volatile sensor for bananas that will work similar to this. And I think it'll work pretty readily because there's a lot of papers published on volatile production by Fusarium. So we're building, I have a team in NC State, the Emerging Plant Disease and Global Food Security Cluster, and the cluster is working to develop the sensors. We're testing them on a platform with a mixture of pathogens in Western North Carolina. We're going to differentiate plant stress from pathogen stress, so biotic from abiotic. We're developing some wearable sensors that we can leave on the plant. They can grow over time with the plant and send wireless signals back to allow us to know whether the plant's done it or if it's growing. And then we're actually developing a database by sequencing all of the data that we're collecting, called Plant A database, and building phylogeny so we can understand what pathogens infect the crop, and then develop spatial models using collaboration with the Geospatial Analytics Center to look at how pathogens spread in space and time. And the idea is to deploy these sensors in real time in the field, and we have a pending PSI GRIP proposal at NC State for the Plant Science Initiative. We're going to try and pull all this together. I'm also do some work on historic outbreaks, and I wanted to tell you about some of the questions we're addressing there, because we're not only interested in modern day lake blight, but historic lake blight. We've sequenced the lineage that caused the famine. We know where it came from. We've looked at the genome size, effector diversity, and we're actually looking at whether these genotypes still circulate. So we have baseline data globally on this pathogen. And we've done the work with historic samples from the 1840s from her plant herbaria that had lesions of the pathogen. A group of us sequenced the genome years back, 2009. This is at the Broad Institute up at Harvard, MIT. And the genome was very large, larger than other phytophthora, and there's a number of effectors that are driving the expansion of this genome. These effector proteins allow the pathogen to overcome host R genes. So, you know, if you have an R gene in a host and you have an effector that's recognized, you won't get disease, but if the R gene's not there, you get disease in a simplified description. So what we did, we went in, we sequenced the genome of historic lineages and compared it to this genome we sequenced at the Broad Institute and modern circulating lineages. And we have highly supported monophyletic clade from the historic lineages. The Broad sequenced genome was in the middle, and we found about 120,000 SNPs that differentiated the older genomes from the newer genomes. So we went in and we looked at effectors, because those are the genes that are responsible for the host recognizing for host resistance. And what was interesting was effector diversity was different in the older samples. In fact, in most of the regions, the AVR genes or aviarillus genes that are responsible for pathogenesis, they were absent in the old samples. And there was an expansion of effector diversity with time, so that was really significant. And we're also tracking lineages globally. This is a historic lineage. We've actually found the famine lineage 150 years later in parts of the world like Colombia, Florida, the west coast of the U.S., and in China, India, all over Asia, Southeast Asia, and even in Japan. Our oldest sample, I wanted to connect back with Japan, is from 1901. When Japan opened to the west, lake blight moved in there, and it was the lineage that caused disease there on potatoes was the famine lineage. So our idea here is to pull lots of information together. We're genotyping, we're developing sensors, we're analyzing data, we're tracking migrations. So the idea is in the future you might have a drone in the field or deploy sensors on plants, collect that data wirelessly, send it to a cloud computer, and build phylogenies of pathogens, build spatial maps of pathogens, and predict risk of spread of plant diseases. And I just wanted to end with showing the cluster faculty, Keyshawn Ways, developing some of the sensors. We have a virologist, Anna Whitfield, landscape ecologist, Rob Scheller, and Dave Rasmussen's working on some of the bioinformatics from the work we're doing. And that, I'll take any questions.
Thank you, Dr. Isen. I'd like to invite questions from the audience at this point. Do you have a question?
The work you're doing is really
It's good.
interesting. I have a couple questions.
Yeah.
There's going to be
you have a handful of different sensors that you've gone through, and I'm just curious what you're using to detect. Is it like molecular sensing? Is it protein? Is it DNA? Is it cell?
Well, with the LAMP sensors, we're going after DNA, so it's DNA based. But the volatile sensors are, when the plants are infected, they emit various different compounds. They're aldehydes, hexanol. I have it's on one of the previous slides, but there's a whole series of these, let me find it here, volatile compounds that are, well, yeah, right there. The sensors pretty much are detecting this panel, but there are more than those volatiles produced by plants when they're infected by pathogens, so we can switch out the panel. And basically, the sensors are paper-based. They're about the size of a pH paper, and they have gold nanoparticles that are on the paper sensors, and they react with the volatiles from the plant. The nanoparticles are linked to a receptor that will detect each of these different compounds. And you can see some, we know the limit of detection with the various compounds, and we know we're basically learning, we're reading the volatile signal when we inoculate with a different pathogen. We run it through a mass spec, and then we can tailor the paper sensor to make it specific for the specific disease.
Yeah.
Okay, so I'm going to ask a practical field question because I do work in Africa. So from a practical perspective for farmers and the like, they need something where any pathogen can be determined. So with your sensors, is there a way to tell between healthy plants, period, and that there might be something going on with some type of disease even if you cannot identify it?
Yeah, well, I mean, basically, we can distinguish healthy from disease baseline data for any, whatever plant we set up. This one's tomato, but you'll get a baseline signal when it's healthy versus disease. Then, we're drilling down into what's there. So say if it's corn in Africa, there are a lot of disease symptoms that are very similar. It might be a virus, it might be fertilizer stress. So the signaling would be different for whatever the specific pathogen is. We're really going into trying to look at a particular host and develop a suite of sensors that will identify the common pathogens on that host. So like on corn, that might be, for smallholder farmers, it might be looking at that set of pathogens, probably mostly viruses, which are a little trickier, and it might wind up going with a DNA or RNA-based sensor.
So in this, at the stage you're at, you can do this with drones and with your sensors?
I can what?
You can do this with drones?
We have not tried it with drones yet, but there's interest. We've generated a lot of interest, and I'm on the student committee in Geospatial Analytics Center, and she's using drones right now to just kind of take pictures of crop canopies and determine whether, and reconstruct canopies and determine, but we have not gotten to the point where we can drill down, is it diseased or not. You might be able to detect fertilizer stress or areas where something's happening, but to really drill down to what's going on at the ground, you have to have a sensor. So we're thinking about that, but we haven't deployed anything on a drone yet.
Related to that, how close would this be to deployment in a field in Africa? So where some guy can walk through and be able to say problem or no?
Well, I was in Rwanda last February with the Gates Foundation visioning an African plant health initiative. They're thinking about deploying technologies in Africa. And basically, right now the technologies that are being used are, CABI has this plant health clinic, so growers are bringing their crops into the clinic. They're getting a visual diagnosis based on, yes, this looks like this symptom, and they're using imaging diagnostics, where they might take a picture of a corn leaf and say, okay, this matches with these images, it could be X, Y, or Z. So where the sensors really come in, I think, would be at the diagnostic labs in these local, through CABI or through research hubs. But field-based sensors, a paper sensor, this is cheap and inexpensive, and you can use a cell phone to read it. So as people go out to the field, extension, I don't think it would be ready for the hands of the smallholders, but it definitely is ready for the hands of the extension, the aid workers working with the farmers to start collecting data. In fact, that's what they're planning to do. Right now they're doing this, they're developing a sensor for wheat rust, Ethiopia, and the CIMMYT group. And it's not based on this technology, it's a different technology, but they're trying to deploy that in a small area, but they want to have it in the hands of the researchers and the folks at CIMMYT that go out and beta test it. So, you know, we're a few years away from that, but I'm definitely in the loop with the folks that are deploying things there, and I go over to Africa quite a bit working with some of the agencies that are working, particularly SIP, International Potato Center.
Sorry, I'm afraid we have to move on next program. And thank you again, Dr. President. Some technology today will be presented by Dr. Shunsuke Oishi. Dr. Oishi is OPI and designated assistant professor at ITPM at Nagoya University. His presentation title is Chemical Peptide Protein Synthesis and Cyclic Peptide Library for Phenotypic Screening. Ladies and gentlemen, please welcome Dr. Oishi.
Good morning, and thank you very much for organizers and sponsors. I'm very happy to share my technology to all of you. Okay, so let me start my talk. So I'm from Nagoya University Institute of Transformative Biomolecules. I'm a chemist. I'm working on peptide chemistry, especially peptide ligation, which is conjugating two peptides or two biomolecules into one by making a covalent bond in between two entities. So by using this technology, key technology, we are applying chemical biology research. One direction is total synthesis of proteins by chemical synthesis. And the other direction is chemical screening, phenotypic screening. So first I'm talking about this briefly. So nowadays you can order peptide very easily, like Amazon. If you type peptide sequence you want, like short peptide, you get the peptide within 1 week. So, but the question is, the problem is you cannot get long peptide, like 30, 40 amino acid residues. It's quite difficult to make by using thread-based peptide synthesis. So then the question is how we can get long peptide or protein by using chemical synthesis. So our idea is make peptide segment by a thread-based synthesis. We can produce 40 to 50 amino acid residue peptide by this technology. The other one, then we conjugate these two by using special chemical reaction. Then we get longer peptide conjugated. So if this is long enough, like if this is like 50 plus 50 and 100 residues, we can call this protein. So to do this, we need special reaction to make amide bond in between two peptides. To do that, we installed special chemical moiety, alpha-ketone acid at C-terminus of one peptide, and hydroxylamine at N-terminus of the other peptide. So then after the ligation, we have homoserine residues. This is not canonical amino acid residue, but this is chemically closer to serine, threonine, and like this amino acid. So this homoserine can be surrogate of these residues. So by using this technology, we collaborated with Professor Higashiyama and Dr. Kanaoka just talk this session. So this is uroprotein, foreign tube attractant, the cute movie like this. We synthesized these foreign tube attractant proteins. So today I talk about this AT-luo synthesis. So this one is secreted from ovule. So this is cysteine-rich protein, so 6 cysteine residues. So refolding for making disulfide bond form active form of the protein. This is 71 amino acid residues. This is impossible to make just by simple thread-based synthesis. So our idea is to to divide this long sequence into two. So leucine and asparagine. So because leucine, we can make alpha-beta acid relatively easily. There's amino acid, hydroxylamine, which will be homocysteine after ligation. So this is chemically crossed to asparagine residues. Then after ligation, we deprotect cysteine protecting group, and then submit it to refolding condition. Then we got active synthetic peptide. So the question is why we need to make protein by chemistry. So yeah, of course you can express protein by using E. coli or other cell system or cell-free system. However, if you want to label protein or modify protein, it's quite difficult. If you put labeling reagent, these are usually targeting cysteine or lysine residues. So at the end of the labeling, you get a heterogeneous mixture of labeled protein. Some of them has multiple fluorescent dye, which is brighter but less active. Most active variant is non-labeled, so which is not visible. So by using chemistry, we can specifically control the structure of protein. So we can put the site selectively fluorescent labeling. So if you choose the residues which doesn't affect the bioactivity, you can label protein without spoiling the bioactivity. So this is the idea of the chemical synthesis. Then we labeled the protein, dual protein, by using chemical reaction. So this is the result. So we dip foreign tube into the labeled protein. Then check the fluorescent imaging. So you can see here, fluorescent is located on the tip of foreign tube, which means receptor is expressed and located in the tip of foreign tube. If you use just rhodamine dye, no localization is detected. So, like this example, we have synthesized a number of cysteine-rich proteins like this. So these are all plant bioactive proteins. So actually, this class of protein, circulated cysteine-rich protein, is playing important role in plant biological system. So this is it's expected that more than 800 genes in Arabidopsis is encoding CRPs. However, only 20 or a small number of factors are identified and characterized. So a lot of these are still uncharacterized, and function unknown, and structure unknown. So to understand biological system, we need to understand all of these. But it's quite difficult to synthesize because mature form and function are not known yet. So then to study, to work on this problem, we think about the chemical screening approach. So chemical screening is very powerful and classic method to study biological systems. So you take some disease or some model system, and then put chemical, apply some chemicals. So you have chemical library, lots of different chemical structure you apply to this system, then you get phenotype. This has been done by a pharmaceutical field many years. Pharmaceutical company has millions of small molecules. However, the screening is already mostly over. It's difficult to have active compound from small molecule library. Our idea is to expand this library to mid-sized molecule, which means peptide or other bigger entity, molecular weight of more than 1,000. Because this class of molecules is known to have high specificity, like biologics, like antibodies. And these molecules are easy to produce, so we can make this by chemical synthesis. So among these mid-sized molecules, we focus on cyclic peptides because these are known to be good binding affinity and stable under physiological conditions. So to do the phenotypic screening by using cyclic peptides, there is 3 difficulties. One is the synthesis. So cyclic peptide synthesis. So cyclic peptide is a very common class of molecules nowadays, but it's not easy to synthesize. So you need optimization, and you need to take time to make one single cyclic peptide. So next thing is the design of molecules. It's not easy to make millions of cyclic peptides. So our capacity is limited to several hundred. So with this limited capacity of synthesis, the question is how we can design a molecule efficiently to have more hit compound. Then third problem is target identification. In the case of small molecule screening, this is always a problem. Once, even if you get very good hit compound, you don't have a target protein. It's very difficult to identify the mechanism of functions. So the first problem, we developed efficient synthesis of cyclic peptide. Standard method is like this. So from the solid phase synthesis, you get linear peptide with a side chain protection. Then a head and tail cyclization of LC terminus and N terminus. Then after this, we can deprotect all the side chain protecting group to have unprotected target cyclic peptide. So this process, the problem is you have to isolate this and this and this. You have to purify each step. You have to optimize the reaction condition of this depending on each sequence of peptides. So then expert of organic synthesis has to work on this synthesis and optimize, and it takes like several weeks to develop the synthesis of each cyclic peptide. So our approach is using a ligation technique to conjugate two different peptides. So we do this technology in the same peptide head and tail. So we introduced alpha-ketonic acid and hydroxylamine like peptide ligation. By using this technology, we can deprotect all the side chain and ligate one pot, then get cyclic peptide deprotected. So this is the desired product. So we only have to isolate this final compound. So it means just one pot manner, and we don't need to optimize the reaction condition depending on the sequence. It's very easy, and this allows us to make cyclic peptide routinely. So we can produce cyclic peptide 5 to 10 cyclic peptide every week. So so we got like so far 300 different cyclic peptide library. This is not big library. So biology people say, oh, 300 is not enough to get hit compound. Yeah, I know that. But for the peptide chemist, this is incredible number. So peptide people say, yeah, 300 cyclic peptide, it's unbelievable, it's very surprising. So they don't really believe me, but we got 300 cyclic peptide. So next thing is design. So we have only 300 different structure, but the peptide diversity is a lot. If you think about 7 more peptide, the diversity a lot more than the Cas needs to be compound. So which is the number of known other chemicals. So how we can choose more potentially active compound effectively? So we focused on the genome. So like secreted cyclic 16-rich proteins. So this class of peptide has important role, and this is secreted. So it's targeting outside of the cell, like cell membrane. We take the sequence for its loop structure facing the outside of the protein. Take this loop structure and make it cyclic. This is mimicking the surface structure of this. As this class of proteins are binding to receptor protein, so this could mimic the binding. To do this, we focus on cysteine residues. So cysteine residues are forming kind of core in the default structure. If you take a look at this, so these two cysteines are not connected directly, but these two sequence in between this sequence in between two cysteines is forming a kind of group structure. So our idea is just very simple. Look for cysteine-rich secreted proteins from the gene database, so which contains a lot of uncharacterized function unknown and really new proteins contained. Then look at just a sequence, primary sequence of these genes. Then take sequence in between two cysteines. Very simple, no biased. Then abstract the sequence like this, then synthesize cyclic peptide like this. So you may wonder, this simple strategy really mimics protein or not. So we, again, I emphasize, we just take sequence from structure unknown proteins. But if I apply this technology to structure known factors, like this kind of well-studied cysteine-rich proteins, if you look at this, so this structure is in between two cysteines, is forming nicely group structure, is facing outside. So, and the other group will be this and this and this. So cysteines are not directly making disulfide, but these sequence in between two cysteines are forming surface of proteins. So our strategy is taking genome information, and then cyclic peptide library synthesis, and then phenotypic screening, just applying cyclic peptides to Arabidopsis seeds. Then we check the phenotype. So as a result, we got several hit compounds. This is just a few examples of that. So this one, cell division is disturbed. So this is root cell. So usually cell become like this shape, but so this is the unusual cell division. So somehow cell division is disturbed. And this one, root growth is disturbed. This is normal. Somehow, yeah, abnormal shape of root is observed. And the other example is stomata development. By using our chemical, we could develop more number of stomata. So we have several hit compounds, and also compared to small molecule screening, by using our cyclic peptide, we saw less toxic activity. So in the case of small molecule screening, often we can see brown leaf and plant dead. But in the case of cyclic peptide, mostly plants are healthy. This is another the feature of our library. Okay. Then after finding the compound, next interest is target identification. So target identification is always a problem in small molecule screening because it's difficult to find modification site. To understand mechanism, you want to put some fluorescent labeling somehow, and also you want to immobilize molecule to affinity gel. But to do that, if you try to look for modification sites of small molecule, it's always difficult. Once you put something on the active compound, you lose the activity, or you start seeing different selectivity. So it takes years of work. And also, if you succeed to get some functionalization by the non-specific interaction, you see lots of noise. So we guess by using mid-sized molecule, these have higher specificity compared to small molecule, so we can decrease noise from caused by non-specific interaction. And also, these molecules are big, so it's relatively easy to find functionalization site. Like this case, we can just install special artificial amino acid which has modification handle. We can just install this amino acid residue somewhere in the cyclic structure. It's very simple. Then, by using ligation reaction, we can put fluorescent labeling here to do microscope fluorescent imaging study. And if we immobilize this cyclic peptide with agarose gel, we can prepare affinity column. And mass spec analysis gives you the information of target proteins. So for now, we saw less noise as we expected from mass spec results. So this is probably due to more specific interaction of cyclic peptide. The good thing is, once we get this compound, this key intermediate, we can make label and also immobilized version in 2-3 weeks. So after finding hit compound, you can get this kind of information within like 1 month. It's very quick compared to a small molecule case. So this is example of fluorescent imaging, one of the hit compound, which is disturbing cell division. This is root cell. This is membrane labeling, and this is cyclic peptide level. Then, if you see this, so baculose is you can see lots of fluorescent, which means cyclic peptide is accumulated baculose in plant cell. So probably, I don't think cyclic peptide just permeate the cell membrane. So this is probably due to endocytosis mediated by receptor. So we hoped cyclic peptide, we are mimicking secreted protein. So we hoped cyclic peptide interacted to membrane receptor. So this is probably what we expected. Okay, so this is affinity purification and target identification. We prepared affinity column, and then we did verification. So far, that cell division factor, we saw 2 leucine rich lipid kinase receptor type protein, and also 2 nucleus protein. So this is very abundant protein. So I think this is just noise of the result. But we could see something relevant to our hypothesis. Okay. So we have 3 technologies: cyclic peptide synthesis, and design of molecules, and then target identification. By using this, we are now doing phenotypic screening with genome-based cyclic peptide library. Future directions, we want to use combinatorial synthesis approach, and then expand the diversity of our compound to like 10,000 compounds for high throughput screening. So this is what we are doing now. And also potential collaboration is we can create focused library focusing on the considering the expression pattern specific tissues or specific like event. So we can create tailor-made library based on expression patterns. So this is kind of potential collaboration. And also we can simplify this kind of protein by using this technique to create smaller, easy to handle cyclic peptide. By using this technology, we have startup now, so Graphite company. Okay. Yeah, thank you very much for today.
Thank you, Dr. Oishi. I know you are eager to answer questions from the audience. Yeah, thank you. And thanks again, Dr. Oishi.
Thank you very much.
this concludes our first session. We will now take about 25-minute break. Please introduce yourself to our presenters and ask them any questions you may have, which brings better imaging and accelerates biotech research. Ladies and gentlemen, please join me to welcome Dr. Marvelous.
Okay, everybody hear me? Yep. So thank you very much for the invitation. It's a real pleasure to be here, and I'm teaching this afternoon, so I can't really stick around too long, but I'm very happy to have discussions later. So what I'm going to talk to you about is work to improve the ability to do live cell fluorescence microscopy and sort of advancing our knowledge base. And before we get going, I'll sort of have some disclaimers here. So I'm a professor at UNC and in the biology department. I'm funded mostly by the National Science Foundation. I also am a fellow and travel in the summer to the marine biological labs in Woods Hole, where a lot of this work has continued. I have many collaborators who have contributed. These are PIs. There are numerous grad students and technicians here that I couldn't really list. I listed some key ones. I'd like to thank UNC, the quantitative biology program, of which I'm a member. We have really great facilities on campus. We have a makerspace and a physics shop and a glass shop in chemistry. The marine biological labs, where I'm director of the analytical quantitative light microscopy course. I'm also founder and president of MISAR Imaging, and which was spun out around this technology, which I'm going to tell you about today. Okay, so technical challenges to discovery and development. So here I've drawn a sort of schematic of an embryo developing from one cell to many cells, and one of the goals of my research is to understand how cell division results in an organism, and so this rapid segregation of the genome occurs over time and results in sort of a complex organization, which is sort of schematized here. One of the issues with imaging is, so here's a movie I took many years ago, and with high contrast, and so you can see nicely the microtubule of the mitotic spindle in this tissue culture cell. But as time goes on, I'm slowly photobleaching and killing this cell. And you can see that by the increase of the background and the decrease of the signal. And so eventually the cell attempts to divide but fails because of the damage that I inflicted on it by actually imaging it. And so the upshot of that is that we can't take high temporal and spatial resolution images over long periods of time. And so what we end up doing is concatenating movies of different embryos or cells to generate a full view of development. And this takes a lot of time, and it took her graduate career, Lydia's. My scientific grandfather is passed away last month, actually, at 98 years old, Shinya Inoue. And Shinya Inoue was many of you may know, a true innovative scientist. He was a biophysicist who was interested in understanding cell division at a time when we didn't understand what the mitotic spindle was. And there were debates about this that went around the world and about what the actual nature of this was. So Shinya realized that the only way to solve this problem was to image it in a living organism. And this is before fluorescence was available. So he developed a polarized light microscope which could image the mitotic spindle, and finally proved that the spindle was indeed this dynamic structure, for which he was awarded the Japan Prize several years ago. He was also, here's a picture of his microscope. He was in the Ripley's, believe it or not. He built his first microscope on the remains of a machine gun left over by the Americans from World War II. Okay, so what I'm going to tell you about is this technology. This is a light sheet. Here is a light source. His name is Phil. This is a cylindrical lens. It is now taking this light source and making it into a sheet. Here's our sample. Her name is Charlie. She is in the sheet being excited, and this object here plays no role in this device. It's just to trap the light. Any questions? Okay. So light sheet excitation is not something that we invented. In fact, it was first described in 1907, I believe. And the beauty of this is that you use a sheet of light at the focal plane. I'll explain this better in a few minutes. So you only excite the sample at where it's in focus. So this allows little out of focus emission, so high signal to noise, high contrast, fast acquisition because of this, and low photobleaching, again, and toxicity, again, because of this. And so this is the technology we wanted to implement. The problem was the things we work on are extremely small and require the highest resolution possible. Existing light sheet microscopes use lowest, or used, I should say, lowish NA detection objectives. And so numerical aperture is a measure of the angle of acceptance of a lens, which is then the angle of acceptance determines the resolution of that lens and also the brightness of that lens. For reasons like, if you want to know, I'm teaching it this afternoon, so you can come and listen. So here we have a basic orientation. So here we have an objective lens that has shooting a laser in as a sheet into your little blob of cells here. And then you have a second objective, which is orthogonal, or 90 degrees to that objective, and it collects the light that is emitted from this sample at a low numerical aperture, low cone of acceptance. The cells are held here magically in between these two things. So the problem is this 0.5 numerical aperture, and the magnification is low. There's a solution from from Nobel laureate Eric Betzig called the lattice light sheet. The lattice light sheet uses a slightly higher resolution objective, 1.4 numerical aperture, but it is still lower than what we normally use. We would never use one lower than 1.3, for instance. So this instrument, you can buy this instrument. It costs over $1 million. This one lens here from Nikon costs $25,000. This lens you cannot buy. It's a custom-made piece. So NSF is not going to give me $1 million to buy this, so I had to figure this out myself with my student Tanner. So it's physically impossible to use high numerical aperture objectives with a traditional light sheet orientation. I'm going to switch over to this side so you people get equal opportunity. So here we have CAD drawing of a very high numerical aperture objective, which we would use on a daily basis. And here at the intersection of the optical axes, this is all to scale, I should say. The working distance of this lens is right there. It's 170 micrometers. This distance is 11,000 micrometers, and so this is just never going to work. So our goal was to illuminate a sample with a sheet of light, and this is to decrease the photobleaching and toxicity. Number two is to use our traditional high NA objectives, which is required for high spatial resolution and nice bright images. And these are oil or water immersion. And this has to use a cover slip based mounting because these lenses expect that. So we invented LIGHT, and it stands for lateral interference tilted excitation. I'm going to take you through that quickly. So lateral excitation is very simply the light sheet orientation. So here we have a mythical cell, here's our detection objective, and we're generating a sheet of light here at the focal plane, and so we're not illuminating any of the out of focus parts of the cell. We do this with a cylindrical lens. This is not some kind of magic. It's a cylindrical lens is like a spherical lens, except for it doesn't focus in one axis. And instead of generating a diffraction limited spot, you generate a line, and this line has a focal depth, which is called the Rayleigh length, and that depth is the S sheet. Okay, tilted. So it's impossible with a perfectly flat sheet, and the reason for that is, in order to generate a focal point, you have to have this ray of light and this ray of light, and all the ones in the middle have to have the same optical path. They can't travel through different materials. So if we had here a cover slip, then this part of the beam would travel through that. This one would not, and that would result in a non-focusing sheet, or something that wouldn't be useful for us. So so we had to figure out how to do this, and we tried several different things, and the simplest one was to actually tilt it, and I'll explain that in a moment. Here's now a depiction. That is a little bitty cell on top of a cover slip, a little green dot. This is our objective lens, and here's our sheet, and that's still 700 microns away. Okay, so here's what we did. We tilt this whole assembly at 3.28 degrees, and the bottom ray of the converging light travels parallel to the cover glass. That allows us to generate a light sheet directly on top of the objective lens, and so use light sheet illumination with our very high resolution objective. Okay, so this is, I'm going to breeze through this. This is just sort of theory behind this. There's an equation which you can use to calculate all these things. You know, you can write that down if you want. And so one of the key things is the length here. And so we need the length to be consistent over this field of view, so that anything here gets the same excitation light as here, so that we can have a measurement of, we can compare these things. And so, so an issue with that is that when you focus light, you generate a very narrow, so to have a narrow focus, you have a very small region of excitation. A typical sort of embryo, like a mouse embryo, is like 100 microns or something like that. So this is not very usable. If you make this a little wider and sacrifice a little bit, you get up to 50, which is more usable. But we stole a trick from a lab, an optics lab in Canada, actually, where we generate an interference pattern. And this is it's a cosine interference pattern that's generated and extends the Rayleigh length of our sheet, the focal depth. And so here's a view of the actual light sheet. We have 4 beams that converge in, and they generate a standing wave interference pattern at the focal point, which is there. And so here's the central lobe, and now we have these side lobes. If we now analyze the intensity of that, so the majority of the intensity, over 80%, is in the central lobe, and then the rest is dispersed in the side lobes. This is clipped up here because we can't even see these if we don't clip that off. So so these are really minimal. Okay, so this is something that as a biologist, nothing ever really works that well because biology is variable. This was our measured sheet, and this is a simulated sheet, a theoretical one. And I was very impressed by I was shocked actually how well this worked. I showed it to a physics friend of mine, and she said, "Wow, that's not a very good fit, is it?" That's just a different perspective. All right, so this was the original prototype we built back in 2017. So this is a standard inverted microscope, a Nikon, now 15 years old. This is a standard camera. There's a laser launch over here. We have a fiber optic that runs around the back and then up through a lens array that collimates the light into a beam, like a big laser pointer. And then we have a mirror here that I got from my graduate advisor. He first used that in the 80s to build a laser-based system. And then that shoots it across here, and that little piece of tape is holding in our lens. And that we make a sheet on top of our very high NA objective. This is now the commercial version. This was installed at UCSD earlier this year, and so Howard Hughes Institute there. And then so this is the fiber optic now and into our little light box, and all that stuff I just showed you is contained in this one little box here. And we have all sorts of controls and other things. Okay, so here's some movies. This is from the Marine Biological Labs in physiology course there. This is the endoplasmic reticulum in a tissue culture cell on a cover glass. This particular person was interested in branching and joining of the ER. And this is a very rapid imaging. These are microtubules in a frog cell that's grown on the cover slip. Microtubules are sort of my favorite thing, and so I do a lot of microtubule imaging. So you can see them grow and shorten. They're also very sensitive. They're kind of like canaries in the coal mine. That if there is a biochemical disruption of the cell, these guys will quit growing and they shrink all the way back. Okay, so here's some filamentous yeast that is a model for plant pathogens and also for how syncytiums work. And we imaged this for I think 13 hours total, many frames, and they were interested in lineaging these nuclei here, as you can see, over time. And Amy Gladfelter, who's the scientist whose lab this is from, gave me a hug after this because she's been trying to do this for over 10 years and couldn't do it. Okay. So this is again a Xenopus cell, a frog cell on a cover slip. This is processed with a super resolution technology called SURF that Ricardo Enriquez in London invented. It's hard to see, but essentially now these microtubules are, if I measure them, they're now about 2.5-fold increased in spatial resolution that we gain by losing 10 times in temporal resolution. And that's his technology there. Okay, so here's just a few more examples. Let's see if I can. Oops. Those are supposed to start. There we go. Okay, these are human cells on a cover slip. These are dividing, these are the kinetochores. This is a nematode, that's a tardigrade, that's another tardigrade, water bear, commonly known. That's a Drosophila embryo, and that's another nematode embryo. And you can take note of the number of frames here for these. It's something that it would be impossible to do in the past. Okay, so Tanner Videro was a technician in my lab and now a graduate student, and he's a co-inventor of this technology. And I think my wife and the rest of my collaborators, we have other technologies that I'd be happy to speak about too if time permits. Thank you very much.
Thank you, Dr. Maddox. We look forward to questions from the audience. Question?
So
Sure.
you talked about the cost of the previous.
Oh, yeah, right.
What's the cost of your solution? Just
Yeah, I'm not really a business person, so I forget to say this thing. Our unit, the base price for it is $50,000, and so and the beauty of that is that you can put it on existing inverted fluorescence microscopes. Now, there's a caveat there. You need a laser. You also need, a camera and other things. If you have the inverted microscope already, almost certainly you have the camera and you have software and that kind of thing. You may not have the lasers, but laser. So just for your knowledge, for your idea, a laser launch with 4 different lasers in it, 4 different colors, is about $35,000, something like that. And then a brand new camera would be about $20,000. And so for another, say, $100,000, you can take your existing microscope and upgrade it to a light sheet. And so that was so from a business point of view, I have sort of a couple of rules that I follow. One is what I call the NIH rule. So NIH funding is in modules usually, and so a module is $25,000. So it's a 2-module thing, right? So I want it to be accessible by people like myself. You don't have to have it in a core facility. Number 2, I have a rule called the undergrad rule. Undergrads are free for me to do research, and so I need my instrument to be able to be used by an undergraduate. And so we can teach an undergrad to use it in a couple of days, and then they can whack out a bunch of data for us. And so those are my 2 sort of criteria: the undergrad rule and the NIH rule. Oh, I have one more: the 5-minute rule. I'm a very impatient person, and so if I'm talking to a student about an idea, I want them in 5 minutes to be looking at data about that idea. And so that's my 3rd rule, the 5-minute rule. So we design our instruments around those sort of criteria. Thank you for asking that question.
Have you applied it to FRET analysis yet?
Yes, we have. So FRET is fluorescence or Forster resonance energy transfer, and where you excite one floor, it then transfers its energy to a different floor, and if they're very, very close together, they have to be 10 nanometers apart or so. And so it works for that. It's fine. You do with a standard sort of cube arrangement. We've also done we've done some localization microscopy, so single molecule super resolution imaging that can get down to, say, 20 nanometers or so resolution, like it's called a storm or a palm. The other thing I'll mention just briefly is that it's compatible with other types of microscopy. So if you have a confocal microscope, for instance, you can still use your microscope as a confocal with our unit on top of it. Here, I sound like a salesperson. Sorry. Yeah. Any other questions? Thank you very much. I appreciate it.
Our final presenter is Dr. Jack Wang from NC State University. Dr. Wang is Assistant Professor and Director of the Forest Biotechnology Group in the Department of Forestry and Environmental Resources.
Yes.
His presentation title is Strategic Engineering of Populars for Bioenergy and Bioproducts through Multiomics Integration in Lignin Biosynthesis.
Perfect. You're fancy. Can everyone hear me okay? Awesome. All right, so thank you first to the organizers for the opportunities to be here today. Is very exciting. So out of all the audience here, who has experience or have worked with trees before, specifically forest trees? Wow, it's bigger than usual. This is exciting. So the Earth has approximately 550 gigatons of carbon. So imagine this square representing all the carbon on the Earth. Animals captures about 2 gigatons of carbon. By the way, that little red dot at the bottom, that's us humans at 0.06 gigacarbons. Now, if we include fungi, protists, bacteria, viruses, and archaea, then we are at around 100 gigatons of carbons. And then the rest are all plants. Now, out of all of the plant carbon, 70% of those exist as trees. So trees are by far the most abundant biomass on the earth. And guess what all of those carbon are stored as? They're predominantly stored in the form of wood. Now, wood is a wonderful raw material. And in addition to being the most abundant biomass on Earth, it also has been contributing and facilitating and supporting the daily lives of humans for thousands of years. We use wood as fuel for energy, for timber for construction and fine furniture, you know, the building we're sitting on, and for paper to write on your meeting agenda. Now, as Black Fridays are coming up, we order a lot of packages online, so all of the boxes that those packages come in, all made out of wood. And the fibers of wood are also now used in textiles, rayon in particular. And combustion of wood is used to generate electricity that runs cities. So given the wide application and the versatility of wood, what gives it that property? Well, it really comes down to the chemical composition of wood. So on your left here, you have cellulose and hemicelluloses. These are your energy-rich polymers of carbon. The cellulose and hemicelluloses is what you use for pulp and paper, to generate biofuels, and is what is used to make textiles for fabric. And on the right here is lignin. Now this is sort of the nasty polymer inside of wood. This is the protective barrier inside of wood that prevents us from accessing all of these energy-rich polymers and fibers. So in the industrial processing of wood, the removal of lignin is the most expensive, is the most toxic, and it is the process that we try to minimize. So for the past decade, we have embarked on a very ambitious project. A project with a single aim of understanding the genotype to phenotype relationship in wood formation through all the different levels of molecular regulations from the genome, transcriptome, proteome, metabolome, and phenome, to generate a mathematical framework with predictive inference that allow us to strategically engineer specific types of trees that produce wood for timber, for biofuel production or chemicals in general, and for pulp and paper or your fiber production. So I'll now present the base model. So we realized early on that the formation of lignin inside of wood is sufficiently complex that it is almost impossible to understand the consequence of turning on or turning off specific genes on changes at the transcript level, changes at the protein levels, and how those perturbations affect these 25 traits of ecological and economic values. So then we designed this systems approach to understanding the regulation. And I'll go through, and this is a brief overview of what we did in these past 10 to 12 years. So we first generated around 10,000 transgenic trees, and then analyzed systematically changes in the RNA, proteins. We did over 200 enzyme kinetics, and then generated a whole bunch of phenotypic data. And then recruiting engineers, we coded all of those data into a mathematical framework with predictive inference that allow us to guide the genetic engineering where we say if we turned off or on specific genes, how is that going to affect these 25 traits? And you'll see how important that is towards the end of this presentation. So this is what we did, step by step. We first generated the 10,000 transgenic trees. And boy, did that take a long time. So we use a systems that we developed using RNAi as well as artificial microRNA to downregulate the expression of individual genes, gene pairs, and gene families. In that sense, we generated all of those trees. And then we selected 221 lines with distinct levels of gene perturbation and characterized the transcriptome using RNA-seq. At the protein level, we developed a method called protein cleavage isotope dilution mass spectrometry, which is essentially generating stable isotope-labeled surrogate peptides, spiking that into the protein extracts of our transgenic and wild-type trees, and then analyzing it by mass spectrometry. And in that sense, we could quantify the absolute abundance of the enzymes inside of those trees. And then a whole bunch of us researchers got together, sat down and spent 3 to 4 years making recombinant proteins for all the pathway enzymes, and then characterizing over 100 Michaelis-Menten kinetic parameters. And this represents the most complete kinetic profile of a plant metabolic pathway to date. And then for all the transgenics and wild type, we also characterize 25 traits of value. And so, for example, wood composition here, where the black bars represents the variations of these properties in the wild type, and then the red bars represent the variations in our transgenic trees. And as you can see, we were able to modify these traits significantly beyond the amount of natural variation you see inside of the tree. And that is really the unique benefits of gene modification. For example, the lignin content, that nasty stuff, that protective layer inside the wood, in the wild type is about 19 to 25%. In our transgenics, we can reduce that amount to as little as 9% of the wood, a significant reduction. We can also significantly improve biofuel production from wood. We're now, the red dots are for the wild type and the black dots are for the transgenic. As you can see, the biofuel production efficiency in the wild type trees is about 25%. In our transgenics, we can increase biofuel production to almost 100%, a 350% increase over the biofuel production efficiency inside the wild type. And so we generated this array of systems and data, and then we recruited engineers, we pieced all of them data together, coded them into a computer program, and then sort of generated this computer game. And inside of this computer game, we can say now, how predictive is it for us to guide the genetic engineering of trees? So we went back to our transgenics and wild type, and we tell that computer program only the expressions of genes and nothing else. And then we ask the program to predict what the protein concentrations is, what the metabolite looks like, what the metabolite concentrations are, and what trees does these have, purely only based on the expression of the transgenes. And then the model predicted what those values are. So on the x-axis, we have the actual experimental data measured from the transgenics and wild type, and on the y-axis, this is what the model predicts. And as you can see, the model was able to capture a majority of the variations purely based on the expressions of the genes, with an average R-squared value of around 0.7. Some, such as the inter-unit linkages, can predict with accuracy of over 90%. So then what do we use this model for? Well, the unique benefit is that we can now, without spending 2 to 5 years generating a transgenic tree, go into the model and say, we need to reduce lignin content. What is the best genes to modify? And then the model can go and simulate tens of thousands of different combinations of gene editing or gene modification and tell you, this is the gene that you want to turn off, or this is the gene that you want to turn on. But the benefit doesn't stop there. The true benefit of the model is that it allows combinatorial improvement of multiple traits by multiple genes at the same time. Now you're looking at tens to hundreds of different thousands of different combinations of genes that you can modify. So what is the best approach? For example, here, if you say, I want to simultaneously improve density, biofuel production, the ratio of sugar to lignin, and then growth. What gene or many genes do I want to modify to achieve simultaneous improvement in all of these traits? And then the model will simulate different combinations and tell you that you need to downregulate or CRISPR edit this PAL and this CoA-OMT gene families. We will never know that without the model. Even better, if we tell the model now we can simultaneously model 3, 4, 5, or 6 different gene or gene families, what are those combinations of genes that we want to modify? And then the model can simulate those tens to hundreds of thousands of different combinations of genes and tell you precisely what genes you need to turn off or on. So let's look at how we use this model in real-world applications. Oh, but before we do that, I jumped the whole chapter, I'm sorry about that. So, that was 10 years ago. Since then, we have generated a version 2 of the model. And this is currently unpublished. So in the first version, let's recap, we take the transgenic knockdowns, transcripts used to predict proteins, and then use to predict the metabolic flux, and then use as basis statistics to predict the 25 traits. For the past couple of years, we have elevated the model in our generation 2 by incorporating indirect effects. That is, when you knock down the expression of a gene, how does the expression of other genes, or how does the expression of other proteins change? These indirect effects also has significant determination of the predictive power of the model. So we capture these indirect effects. In addition to replacing the simple linear regression, we are now using machine learning, artificial intelligence, and just happens the algorithm that best suits the model is one they called random forest. I love that thing. So how does this new version 2 model compare with the version 1? Well, we were pleased to see that across all 25 traits, Model 2 shows significantly lower SSE compared to version 1, suggesting increased predictive power. Now we can talk about the fun stuff. So that was the boring stuff. Sorry for boring you guys with that. Biomass conversion, what do we use the model for? So we've been working with Dr. Kelly's lab from the hypothermophile research group. So what they did was they found a type of bacteria that can feed off plant biomass, that can feed off wood. But this bacteria is extremely picky. In fact, Dr. Kelly has been going around looking at isolating different types of plant biomass or transgenic plant biomass, feeding it to the bacteria, and see the extent to which the bacteria can convert those plant biomass into biofuels. And most of them converted with very low efficiency, around 25%. So then we teamed up with Bob Kelly and said, can we use this model to engineer a particular type of wood that is compatible, strategically paired with the bacteria that he has, that allows for improved efficiency in biofuel production? And this is the result. So as a control, we first feed his bacteria Avicel, which is pure cellulose without any lignin. And the bacteria can convert it to biofuel with this efficiency around 90%. We then feed his bacteria the wild type poplar trees, and then that can convert it with around 20%, 25% efficiency, which is not very good. It's about the same as when you try with other plant biomass. But when we feed the bacteria transgenic wood that is designed specifically for the bacteria, the bacteria can actually convert it with the efficiency that is identical to that of Avicel pure cellulose. So essentially, we have generated a tree that grows in a greenhouse that has the same biofuel conversion efficiency as pure cellulose. I mean, we were blown away by the results. It's very exciting for us. So picture speaks 1,000 words. This is what it looks like. So as control, we have wild type, before and after. This is without bacteria. And with bacteria, as you can see, really not much different here. Now, our transgenic wood, before and after, without bacteria, you see some degradation because the trees are less recalcitrant. But if we add the bacteria, as you can see, the bacteria was able to almost completely hollow out the wood, releasing all the sugars inside of the cellulose and hemicellulose and converting those to usable biofuels. And this is another image showing that for the exact same biological treatment, adding the bacteria, incubated for a week at elevated temperature, the wild type trees, very little stability. And then the transgenic trees, we can see very turbid, releasing all the sugars inside of the wood. So building on these exciting results, we have recently launched TreeCode, which is NC State University's latest startup company to hope to bring this innovative technology to the society. So this is our team. On the left here, we have the tree people. Ron Sidorov, which is a distinguished professor at NC State Emeritus, and awardees of the Marcus Wallenberg Prize, sort of equivalent to the Nobel Prize in Forestry. Vincent Chen, who is pioneered the research on lignin biosynthesis. Me, in the middle, you don't have to worry about this guy. And then on the right, we have the CRISPR pioneers. Rodolphe Berenguer, which many of you guys know, member of the National Academy of Sciences, member of the National Academy of Engineering, and really the person who discovered the function of CRISPR. And then his good friend Charles Gerback, which is also a pioneer of CRISPR and has been using CRISPR very successfully in biomedicine and therapeutics. So this is what CRISPR brings, and this is the value of TreeCode. So TreeCode aims at increasing the breeding cycle using a safer CRISPR technology to increase genetic diversity and meet the regulatory path. So these are the benefits of CRISPR engineering. And in terms of forestry, the model, as you've seen, facilitates target selection, which trees to edit using CRISPR, the design of CRISPR, delivery, and then regeneration, and then most specifically, importantly, improvement of commercial trades for fiber, for biofuel production, for pulp and paper. So this was 10 to 12 years of work, and couldn't have happened without all of the awesome collaborators we have globally. And specifically, these two young gentlemen here, Vincent and Ron, who has been working with me, guiding me on this research. Thank you. So with that, I'll take questions.
Thank you, Dr. Wong. And we look forward to questions from the audience.
Yes.
So among the 25 traits, how many of them are complex traits?
So, how complex? So if you are talking about, so some are obviously more complex than others, such as growth and stem diameter and biofuel production. Those are all complex traits determined not only on wood formation or the properties of wood or lignin, but it depends on the contribution or the level of productivity that a particular modification has on a trait. So with the current model, version 1 can capture about 70% of the variation generally across the 25 traits. Version 2 can capture even more variation. And we're now working on model version 3 already, which is the incorporation of higher level interactions, protein-protein interactions, and then protein metabolite interactions. And the hope is those interactions will further improve the predictive power. So we're presenting, so this is sort of like a base model that predicts certain traits to a particular predictive level, and then incorporation of additional levels of regulation will further improve that.
So do you see the complexity that because you do have a large set of data,
Right.
so do you see the complexity of data help you to increase the prediction power, or actually you are now still?
Absolutely. I mean, as we move towards machine learning based algorithm, you know, the more data, the more we're able to train, segregate, train, and retrain the model to identify not only higher order regulatory components, but also to fine-tune the model parameters that improves productivity. So the limitation for us here is the generation of transgenic, or as we move towards CRISPR-edited trees. So tree transformation is notoriously difficult, and is something that we have been working on over the past 20 years to tailor the design to optimize. And to date, we have a very efficient tree transformation system where we can generate transgenic trees now in as little as 60 days, 2 months, compared to the normal 2 to 5 years of generating transgenic trees. So that has really facilitated and accelerated the production of transgenics that allow us to validate and then this model, and then to generate new variants based on the model guided strategies.
2 quick questions. Have you looked at the adverse effects, disease, resistance and things like that? Because the lignin is there to
Right.
And also, how much of this data is going to be transferable to other species other than populace?
Great question. Okay. So the only physical traits we have included today in that list of 25 is growth, as in height, stem diameter, and estimated stem volume, as well as the density and mechanical strength, modulus of rupture and modulus of elasticity. Because for this first model, everything is done in a controlled transgenic approved greenhouses where we maintain uniform temperature, disease-free, and in a very constant condition. And this gives us the benefit of limiting the variation we observe to genetic effects as opposed to environmental effects. Moving on, field test is definitely the next logical option where we can begin to see and incorporate the effects of the environment, biotech abiotic stresses into the model to further capture these variables. So that's definitely, you're spot on in terms of the next logical step that we're taking moving forward.
And transfer to the species?
That is in the works. So we have looked at lignin biosynthesis in many different species today. Not only popular Trichocarpa, most species, but also hybrid populars, sweet gums, loblolly pine, radiata pine, etc. Now, the extent to which this model is transferable, we really don't know. And that is, again, due to the limitation of genetic engineering in trees, where we don't have an established transformation system for most of the tree species. But some we do have, and those are, well, will be very interesting to see. Now, in addition to trees, of course, all plants have secondary cell walls. So whether the models is applicable to, things like switchgrass to reduce the recalcitrance, improve digestibility for feed, and things of that nature, that is also to be worked on. But there's great potential to build on using knowledge that we have from the model. Oh,
I have another one.
I'm popular today.
Is the modified lignin, you've heard the quote, of course, you can make anything from lignin except for money. Is the modified lignin going to have potentially a higher value because you can now extract it more cheaply than actually makes them.
Well, so lignin, the value of lignin, to now really is just burn it. But the difficulty and the limitations of the lignin wood primarily is due to its recalcitrance from access to cellulose and hemicelluloses. So what we are trying to do here is reduce the recalcitrance by modifying lignin, thereby not only reducing the amount of chemicals, heat, and enzymes required to release the sugars, but also to remove or reduce and minimize the environmental consequence of industrial processing. So something that I didn't cover here due to time limitation is that the industrial processing of wood is one of the major contributors of greenhouse gas emissions. And then combining that with deforestation contributes almost 18% of the global greenhouse gas emission. So by modifying lignin, we can reduce greenhouse gas emission. By making the trees more valuable, you can reduce deforestation. So that is what the goal of TreeCo is. Thank you.
Sorry, I'm afraid we have to move on. Thank you again, Dr. Wang. Now we will shift gears again to the panel discussion how we can promote industry and academia collaboration and regional economic development. We have 4 distinguished panelists here today. The discussion is moderated by NU Tech Director Tomo Sakoyama. Sakoyama, mic is yours.
Good morning, my name is Tomo Koyama. I'm director of NUTech. Next 40 or 45 minutes, we would like to do the panel discussion on how we can collaborate, promote collaboration between academia and industry. So maybe this side is very difficult to see, and first of all, I have to apologize. This uncomfortable stools. Yes, let's move on. First of all, I would like to give you each 1 minute to introduce yourselves, your name, and what you do in the organization. So first, John. Mike.
Good afternoon, everyone. My name is John Loyak. I'm with the Economic Development Partnership of North Carolina. We are an agency that works very closely with North Carolina Department of Commerce to bring in new business into the state. We promote visitor spending and tourism here in the state. The 3 groups that I manage, though, are all focused on existing industry. So that's where we will focus a good bit of our comments today. So I thought I would share with you all that the state actually does a number of different things to support existing industry, to help with small businesses, a variety of things that we do to counsel them and get them to grow, and international trade.
My name is Akihiro Saso, Vice President of Nagoya University. I'm in charge of academia, industry, government collaboration. Personally, I'm a professor of engineering. My major is aerospace engineering, fluid dynamics and propulsion.
Good morning, my name is Jörg Bauer. I'm responsible for Scouting Collaboration Management at BASF. We are here in the Triangle Park with Agricultural Solutions. And beside Agricultural Solutions, I'm responsible for bioscience, so this is everything biological in BASF, so also responsible for industrial biotech and toxicology, so very important.
Hi everyone, I'm Kuntaran Chauhan. I'm a Director of Licensing with the Office of Research Commercialization at NC State University. So our office manages all IP developed by NC State employees, so that includes faculty, researchers, staff, in some cases students. So we perform 2 functions at a high level. There's a compliance side and there's a commercialization side. And the compliance side, it is, as inventions are developed, there's generally some funding associated with it. So what are the contractual obligations associated with that funding? So for example, if it's federally funded, there are certain federal regulations that you have to follow. If it is industry-sponsored research, then there are terms in those agreements that, again, we ensure the university is compliant with. On the commercialization side, our key goal is to help transfer or facilitate the transfer of early-stage research happening in the labs to the marketplace, and we do that through licensing. So we build an IP strategy around technologies that are developed at the university, and then we license them to existing companies. So again, it could be existing companies, big or small, or it could be startup companies that are formed to commercialize.
So we have two from academia, one from industry, and one from public sectors. So it's a nice combination, I think. Today I prepared 4 topics to talk with. First one is the importance of academia-industry collaborations. And second one is current activities and strategies. And the third one is a successful case as a result. And the fourth one is key role for each player to further promote collaboration. After 4 topics, I would like to open the mic to all of you for questions. So please think about your questions during our questions, discussions. So first topic is the importance of academia-industry collaborations. I think all of you agree collaboration between academia and industry is most important thing to bring innovation to the societies. Here I'd like to ask again to each player why collaboration is important from each point of view. So maybe first, John, what impact such collaboration bring to regional economies, and also why RTP is very special as a platform?
Sure. What I would say is, in my line of work, in economic development in general, collaboration is not something you hope for. Collaboration is a requirement. My boss, as he travels around the state and around the world, talking about North Carolina as a place to do business, he is quick to remind people that this is a team sport. This is something that no one organization does alone. Even from within the economic development community, you can imagine if you're doing a project here in Durham or Wake County, you've got somebody from the state who's supporting you, you've got somebody from the county, maybe the city who's supporting you. And so if all those pieces aren't humming along to the same tune, it's noticed by your clients. And so when a company like BASF is looking for a new location or looking to expand, they know that they will certainly do a lot of research on data. They'll know what populations look like in a given area and what the overall workforce will look like today and in the future. But they also know what kind of activity exists in between business, in between the government, and between academics. And it's very quick or relatively easy to tell when the chemistry is not quite right in between some of those organizations. And so one of the things that we strive to do here is to make sure that everyone is at the table. We're going to talk about some of the examples, some specific examples that we've seen this year. And I can promise you some of those are very, very direct examples of the type of collaboration that exists. I have not planted some of my partners in the room, but I have several partners that are in the room today who are working on these projects with us. Michael Bussell from NC State is here, and he and I were talking earlier, and the 2 words that he used as we were kind of talking about a similarly related subject are respect and trust. These are the types of things that have to exist, and it's something that we have built on here. It's something that we will need to continue to strive to improve as we move forward. But that really, I think, is a very important part of it, part of the overall, one of the ways that you achieve the type of collaboration that you're going to need. The one thing that I can promise you is that economic development is very easily, and the person sitting in front of you today is someone who has worked for a software startup. I've worked in medical devices worked in the pharma industry, and I can promise you, those are all very competitive industries, but all of it pales in comparison to economic development. So folks trying to bring business in or trying to get existing industry to expand know what an amazingly competitive world it is. And so you need to make sure that you're bringing everything to the table. That includes researchers from your universities, that includes engineers from the Department of Transportation, that includes a specialist who can provide customized training from one of our community colleges. All of those people, you have to put your best foot forward every single time you go after expansion or a new business. That's a big part of what we do. We are blessed, certainly, in that we sit here today in the midst of the Research Triangle Park. This is, for those of you who don't know much about the history of it, this is the year that we are celebrating the 60th anniversary of this park. This is something that took some fantastic vision 60 years ago, but it's taken a lot of dedication to continue to improve it. And it's something that, John Pisa, when you asked me what one of the strengths were, I would say it's the reputation of this park. This is something that when people think about North Carolina, internationally speaking, I love the state of North Carolina, but I can promise you we do not have the brand that Florida or Texas or Chicago, New York, and Los Angeles have. But the brand that they do know, besides Michael Jordan, is RTP. And we had a delegation that came over from the United Arab Emirates back in August. And we took them here, we hosted a luncheon here at the Biotech Center. And this group, as they met with folks from around the area, were introducing their own research park that they're developing in Sharjah. And they referred to RTP as the grandfather of the Research Triangle Parks. So we are blessed to have what we do, but we need to keep making it better.
Thank you, John. I like your words of team sports, and very interesting, and you take proud to be your member. Yeah. And maybe next some industry persons, why do you collaborate as academia, and then why your office is located in RTP?
I mean, it's, as we heard before, it's a very obvious question. Collaboration is absolutely a must. I think from a company point of view, we all think we're the smartest, and I think that the first realization is that it's not the case. You have to reach out and find people who are smarter or and can help you. There's a lot of technology is moving so fast nowadays. There is no way to stand that alone. You can spend a lot of money and you will not be able to get there. Specifically at BASF, I mean, for us, this is ingrained in our DNA. I think we have 154 years of success with collaboration, so we can say this is a model which is working for us specifically. Why are we here in the RTP area? To move on to that question, it's very logic, actually, very logic. And when we celebrated the 35th year of the NC Biotech Center here, the story was told how the president at that time went to the Rhine area because they realized that all the big chemical companies were at the Rhine in Germany, and they could attract these companies to come here, and I don't think anybody regretted it. So it's a great place. There is, I think, talent is what would be for me the first point which comes to mind when I think about RTP, RT, talent, P, very good. So I think with the universities here, with all the companies here, this is a very good area to do science.
Thank you. This is very obvious question. They said, Dr. Sasso and Carlton, would you share your thought?
Yeah. First of all, I see similarity between RTP and Nagoya area. Here, there are good universities, academia, and industry, in particular in biotechnologies. But in Nagoya area, we have companies in different fields, automotive, manufacturing machines, and aerospace. For example, Toyota company and ICAW and Denso and so on. And also we have academia, and we believe that the Nagoya University is a leading university in that area. And so Nagoya area we have companies, industries, and academia is clustered within a circle with the diameter of something like 20 miles. So we have a good access between academia, among academia and industry. And I believe that, already you mentioned that to companies, on company side, now the technology is advancing very fast. So the company has a lot of program in the interdisciplinary sense. So in that case, the one section in the company and in the company itself cannot solve the problem alone. But we academia, we have many the schools and many research fields, engineering and agriculture, and of course biotechnology and so on. So we can form the groups of researchers in a very flexible style. And on the university side, because the about 80% of research budget it comes from a governmental project. But as you know, the budget is limited from the government overall. So we have competition among universities in Japan, but the resources are limited. But on the other hand, we have room to increase the research budget from the companies. So in that case, we try to triple the amount of collaboration with the industry for the coming 5 years. So in those senses, we have a mutual benefit to work together.
Thank you. Dr. Sasso mentioned, I totally agree, Nagoya is very similar to RTP, industry and academia. And today we have Chiyoko-san from Japan Societies. I heard she is working also looking for sister city or Lori or RTP. So Nagoya might be a good fit in that sense. But creation is quite different, but that might be good.
So just to add to what was said before from a university perspective, and particularly from NC State's perspective, I think it's really critical to be collaborating with industry in terms of meeting some of our mission goals. So one critical goal for university, right, is to produce the next generation of talented leaders, entrepreneurs, and understanding what the needs are of industry is critical. So understanding what the industry needs, making sure we have programs in place to help facilitate that, so we're attracting the right talent to the region, we're educating them, and they're then staying in the region. So that's a critical part. Initially in my introduction, I talked about commercialization. So a critical thing that we do at NC State is research commercialization. Now we're good in terms of doing early discovery, identifying technologies, building an IP strategy, but we don't, as a university, we don't make products, right? So we have to find a potential partner who's willing to work with us, get them excited about the technology, they're ready to invest in that technology and further develop it. And if you do that properly, right, you can bring more products to the market. Again, from a university perspective, it's bringing products that would benefit the consumer. So bringing those to market, that's going to help create jobs. Economic development can be a big aspect of that. The key thing is also bringing venture capitalists, right? So at NC State last year we did 21 startup companies.
a key aspect or thing to get those further developed is really venture funds. So creating these companies, the startup companies, making sure you have bigger players in the region is all critical in terms of developing the region and also making sure we're relevant.
Thank you. I think after all comments, we all agree collaboration is important. Of course, this might be an obvious question, as you said. And as we move on second topics, as the second questions, I would like to specifically ask the academia side exactly what you are doing with collaborations, typical activities and strategies, which is Bertrand, and after that, Dr. Sasso.
So that's a great question. I think we don't do enough in terms of marketing ourselves. A lot of collaborations are passive. A lot of them are, it's publications out there. It's our PIs going and talking at conferences. But I think in the last few years we're starting to realize that it's really important that if we're doing exciting research, we need to be talking about it. We need to be actively trying to find collaborators. So, for example, at NC State, we have building communication teams. I just found out today Cal's has a communication writer too. So we're trying to bring in people who can talk about the research happening at the university, and also trying to make sure that the system, the process is as straightforward as we can make it. So at NC State we have a partnership office, which is a key contact. So if industry is interested in collaborating with NC State, the key thing is to understand how do they want to collaborate, like what's that type of engagement? Is it because they want to get access to talent? Is it because NC State has great expertise or facilities, they want to do certain type of testing? Is it because you have, NC State has technologies they want to license? So having that first group that can talk with them, identify how they want to collaborate, and then bring in all the relevant parties. So we have the partnership office. I know colleges and departments are also now bringing in, I'm just going to loosely call them ILOs, this is industry liaison officers. So these are individuals who understand the expertise within the department and college, and then are interfacing with industry, so as with the goal to make matches. Right? So if industry has a question and they would want to get access to NC State, they might not know who that right PI is, or whether NC State has that facilities or not. So that person would be a good place to start. Similarly, as you have faculty at the university coming up with new ideas and want to find a partner, this person would be a great contact to go to and see if they have within their network people that reach out. In addition to that, even our office now, we've hired a communication, a digital person. Again, the goal is to try to emphasize on marketing, so we can talk about the technology that we're developing at the university to actively build collaboration.
Dr. Sasso?
I can introduce two special schemes doing by us. One is what shall I call organization to organization research, joint research. Usually, the collaboration with industry is done by a single research group in university and a single section in a company. But in that sense, we can solve the program in a very specific way. But as I say before, the program becomes more complicated and multidisciplinary. So we group by plural research groups in university, and counterpart in a company is consists of plural sections. So in that case, we conduct the research, collaboration research, section to section, between section to section, but in 2 times in a year, we have overall total group discussions and review meetings. So in that case, all the players and researchers and the people from company got together, including executives from the company, CTO, and vice president like me from the university side. So in that case, we can discuss. We listen to the presentation of each specific project, but we have time to have a discussion for overall. So in that case, we can strengthen the collaboration in between. Another scheme is to have a laboratory of company in a university. So we can offer, yeah, we got the money for that, but anyway, we provide a room, closed room, secured for the secured, and then overall the standard area is some 2,000 square feet. So in that case, company, and so we invite a designated academia from a company, at least by two of them. Then the company have a good connection to university, and also company has a very flexible freedom how to conduct research work. So the research in that special lab, the company itself alone can get the IP, and also they have a choice to collaborate with university, academia. So depending on the commitment of each specific research. So in that sense, we can strengthen so we have a mutual benefit to work together closely in a common field. And also, as a starting point, we have a special the light scheme. In that case, area is 500 square feet. Then we can collaborate. We can start with a small project by a small room and a small number of the researchers and the academia, so that we can see how this project grows largely or not. That's it.
Thank you. Carl Tran and Dr. Sasso introduced their activity on academia end, and now I would like to ask the industry side. And how do you collaborate with universities? And do you think there is any difference collaborating with US universities and collaborating with foreign universities, including Japanese universities?
Yeah, so I mean there are a number of different collaboration models, and for instance, we at BSF we have research alliances where we have specific places where we actually have dedicated people at the university. Here in North America, that would be, for instance, in Boston or in California, at Berkeley. We have focus universities where we have master agreements running here with NC State, for instance, which we recently extended. And then, of course, you have the more bilateral. So a researcher sees interesting scientific work and then tries to reach out. So I think these are the different models. They all are based on, I think, three fundamentals. The first is the relationship, and we heard that in the beginning. It's kind of like it's like dating. You know, you have to build up this relationship, and so I think both sides have to see what everybody brings in. So it's kind of like there's still the thinking that industry has pockets full of money and they don't know what to do with it. And then on the other hand, there's the thinking that scientists just want to publish and they don't care about IP or whatever. So I think in that respect, you have to build up this relationship. The second point is kind of like the feasibility. Does it work? And I think NC State is a great example where they realized that you cannot make it too complicated. You cannot, put all kinds of things up there. You have to be pragmatic to get things working, and this is also an important thing. You cannot have everything for yourself in a collaboration. You have to share. To be pragmatic. And the third thing is kind of like, this goes back to Paul's 5-minute rule, is I have to explain it in 5 minutes if it's interesting to get budget for it. And if I cannot do that in 5 minutes, then there is no budget. So if I compare that now with our interactions in the US, in Japan, we are also very active in Europe, of course. All these places have different needs and sometimes also rules. You have to adhere to it. Take some understanding, also some cultural understanding. I think one thing which for us in the interaction with Japanese companies is obviously as difficult as the time difference. It's kind of like to have a phone call at 5 in the morning or at 8 at night is usually, you need a couple of good jokes to keep things going. And so that's one thing. It's also, I think we are very fortunate that we have a person in Japan which helps us also to make these interactions, because that is also what we realized is you need somebody who speaks the language, who understands the cultural needs and also gives back a sense of how long things take and what are differences. So,
And thank you. I'm sorry that this question is not included in this scenario, but you mentioned a master agreement with NC State University. Such kind of master agreement, what is decided in that agreement?
So the master agreement actually, and we have that also with other places, but not too many, I have to say. So NC State is we favor them. The idea is really to avoid this bilateral negotiations. So, as I mentioned, one form of collaboration is, okay, somebody sees something interesting, a publication reaches out to the university, there might be not that much contact, so you go through then 3-6 months of sorting out how would an agreement look like. If you have a master agreement, you have a template. So you don't have to spend this 3 to 6 months. You know how, in general, things are going. You have a CDE already included, so you don't have to do all these kind of things in the background. So it makes it much faster, and then it helps also both the scientists in the company and at the university to talk faster about science and not spend too much time about all these legal things.
Yeah, we love to conclude such kind of master agreement. Yes, after we hear the each activities, and now I'd like to ask the next question. The third topic, what's the result? So, after these activities, what result do we have? So, John, maybe to introduce the success case. Which company recently moved to RTP, and what impact does it to RTP?
Sure, I'd be happy to. And of course, I have to start here in the Biotech Center and talk a little bit about the life sciences industry. And so, the Economic Development Partnership of North Carolina, each year we are involved and essentially close about 150 different projects statewide. These are from very, very large projects in Wake County, Mecklenburg County, thousands of employees, to what could be a 10-job expansion in one of our rural counties. But there was one that was kind of special this year. It actually occurred down in Lee County. For those of you who don't know where Lee County is, just get on Route 1 and head south. You'll be there in about 30 minutes. Sanford is the biggest town that we all know in Lee County. And it was because of a research project here at the Biotech Center that Pfizer really began to look at North Carolina as a potential place to expand some of their manufacturing. And so there is a facility down in Sanford. You can see it from the highway as you drive by. Pfizer has a huge sign advertising their presence there. But this year, they came to us because they were going to expand the facility. And so based on a relationship here, based on things that happened here, in this entity that the state of North Carolina began investing in 35 years ago, we are still seeing success come from that investment. And so Pfizer has committed this year, just a few months ago, they committed to doing an additional 300 jobs, which is fantastic for Lee County. But more importantly, it's a $500 million investment in Lee County. This is huge for the area. And for those of you who are wondering what we do for existing industry, just know that it's not easy to move $500 million once it's invested here. And so we at the state understand what that means. We're very, very happy to have it. I'd also have to point to expansions done by companies like Merck, more life sciences expansion here in the area. And what we're seeing is, from the life science perspective, it is expanding beyond just Durham and Wake, maybe Chatham counties. It is, and Orange County. It's expanding into Franklin County and Johnston County and Lee County and other areas. And so the Merck expansion was one that we were very happy to see. Again, a couple of hundred jobs. The other thing that's happening is, RTP, founded 60 years ago, originally started out very focused on information technology. And so over 60 years, one of the reasons that RTP really has seen the success they've seen is because of 3 major research universities. That's really what makes this location unique. And so there's been a lot of focus on information technology through the years, and now life sciences. But the information technology expansion and the effort that we put into that, and all of the things that we're doing with STEM education in the state, whether it's K through 12 at a community college or the universities or graduate and doctoral programs, all of that effort continues to build. And so Charlotte, a few years ago, was really largely known as a hub for finance industry and banking. It was known as a place where one major utility players like Duke Energy were. But Charlotte has kind of very, not so quietly, expanded its reputation as an IT hub. And so more and more we're seeing expansions there. And 2 that I want to call out today were, number one, there's a small company called Microsoft who recently announced an expansion, 430 jobs in Charlotte. And to be perfectly frank, that was one at that level in Charlotte. Got about a day's worth of news. 430 jobs. I mean, we are really proud of that. But it was certainly met with some fanfare. But people almost come to expect that sort of a win for a place like Charlotte. The other one that was really important was the 2,000 job global technology center that Lowe's Home Improvement has committed to out in the Charlotte area. Now, here's what goes into winning a project like this. First and foremost, you have very talented people, consultants, HR professionals who are doing research on whether or not you have the talent. So before they even approach the state of North Carolina to find out how we might support a project like this, research is done to find out if you have that level of talent. 2,000 new employees anywhere in the U.S. is not necessarily an easy task to complete. And so they looked at this and they compared us with places like Nashville, Dallas, Denver, and others. And it was one that we were able to win. And we're really proud of that. And so 2,000 IT jobs in Charlotte, I think, is something that does tie back to the investment that was made here in RTP and the growth that we've seen. And so we've seen some good wins. I will tell you this, nobody is stopping. Nobody is taking any time off. We know that the economy will begin to slow at some point. We're watching all the indicators. But here in North Carolina, we want to see every expansion we can, and we want to see every new business that we can. And I think we've got a great environment for it.
Thank you. Next year, I think BASF has a big international companies. There's many cases collaborating with academias. To share one examples, successful examples, and share why it is successful.
Yeah, maybe 2 examples.
Oh yeah.
Very quick ones. So one is a bee-friendly, natural derived insecticide, Inscalis, which will get on the market next year. This has been developed actually with the Kitosaki Institute in Japan and with a Japanese company, Meiki. So this is a very nice example. Insecticides is not too easy, so this is a good success. The other example is also out of agricultural. This is a product we're marketing together with Cargill. It's called Latitude. It's a plant-grown, long-chain polyunsaturated fatty acid oil produced in canola, and this was also many years of university collaborations to bring that science to a part where you really can produce these algae fatty acids in canola at the end of the day.
Is that any specific reasons, those cases?
Yeah, I mean, as said, in both cases, the realization that other people had great ideas or had a different idea, and that you really identify these and then work it out together and move it forward.
Thank you. Next, academia end. Dr. Sasso.
Two examples which should succeed. One is our Center of Excellence project. First, it's still funded by the Japanese government. The subject is aiming in creation of human-centric mobility society. So in that project, we are doing research work of autonomous driving technologies, and also we are conducting, we are collaborating with several municipal governments to implement the real the demonstration in the technology, and also. The creating a society for human-centric should be taken care of by the health itself. So in that case, one of the important goals for the project is to make the elderly people to live in a healthy sense and in a happy life by using a mobility society. So in that case, in the project, we have some players of nano-life systems. So in that case, they are developing some the sensing systems for the human health, and so and also there some players are developing robotics. For support the elderly disabilities. So this should be, and this project is about $50 million project for 5 years or so. And, but this induces the investment from companies. So we have several company laboratory for that project on campus. And another project is gallium nitride, and we have a Nobel laureate Professor Amano of the same age as me, and he got a Nobel Prize 2014 for the inventing blue LED. But now his project is going to from a light to energy, power electronics. So now they are trying to get a good device by using gallium nitride to save energy systems, in particular for the inverters and so on. So that should be used for electrical cars and even to electrical airplanes. And another very small example of the success is to develop photocathode, which is now invented by a researcher in Nagano University, his origin is from space science. So in the process of detecting some very small signals, and also his interest is turning to developing a photocathode. The photocathode is cathode, not to emit electrons, not by the ohmic heating, not by field emission, but also by eliminating exciting light. So in that case, by controlling the light with a specific wavelength, they can produce a very high quality electron beam. So in that case, this electron beam has a very high quality and low fluctuation, low temperature. So in that case, this is very useful for the electrical microscope and so on. So this is a very and now he's running in the startup company, and in order to make his product to the market. Yeah, this is a very small example.
Thank you, Carlton.
So again, from NC State's perspective, I think to just talk through a few successes, every year we bring in close to about 56 million in industry-sponsored research, which I think is testament to research happening at the university and the flexibility that we have developed over time in being practical when working with research partners. Just quickly about statistics from our office. So last year we got about close to 300 invention disclosures. We filed 100 new provisional patents, so it's not talking about PCTs or national stage filings. We did 150 licenses. Out of those, 42 were for utility patents, and 50 were for plant varieties. So these are plant varieties developed through traditional breeding that are protected generally by plant packs and PPPs, and we did 21 startups. Jack talked about his startup, Trico. We're very excited about that one. But just to give you another example, Locus Bioscience is a startup that came out of NC State in 2015. This is on CRISPR type 1 system, not CRISPR type 2, so it's type 1. And they're using this to repurposing the system to kill human pathogens. So we were formed in 2015. We raised 21 million. I think it was 2017, and in January this year, we just closed a collaboration deal with Jensen for $800 million, and potentially they will go into human trials next year, starting off next year. So that's a great success. We're very excited. We've created over 40 jobs in this area. They're going to expand. So we hope, as we are putting out a lot of startups, doing collaboration agreements. Again, this is early stage research. A lot of these are going to fail. That's just the nature of the business. But if we provide enough support structure, the ecosystem is here. Hopefully, a lot of these will be successful.
Thank you. Actually, we are running out of time, and I have to skip the whole spot. But before opening the mic to you for questions, and I would like to ask one question specifically to John. RTP is the best platform for the innovations, many players, academia industries. How do you envision the future of RTP in near future, in long-term futures? What is the future RTP behind?
Sure, it's actually a pretty exciting time here in the RTP. Quick show of hands, anybody who's worked in the park the last 20, 30 years? Sure. How many restaurants are there in the park? How many of you have lived in the park? So, the commercial and residential side of things has always been lacking. And what I am told by my millennial colleagues is that they're really looking for something that way. And so I think one of the most exciting things going on right now is what's being referred to as Park Center. Center portion of the park, for those of you who knew what the old Radisson Hotel was, is really that kind of, and the RTP had called some of the buildings there the Frontier. We're seeing now new pop-up restaurants that are going to be in shipping containers popping up there. It's going to be residential, commercial, a lot of retail out there, and this is all bordering 40. And so people driving through this area didn't even know that they were driving through the Research Triangle Park, unless, you know, they occasionally would see a sign. And so this is something that really is going to transform a very small portion of the park. There's still a lot of land out of it, but this will essentially be a central hub that will have some of those options that it's never had before. And I think that's one of the most exciting things that's coming.
I agree that we need some central hubs. Many people coming from Japan to see the RTP, so I drive the RTP, but they can't see anything. It's green. So we need something. Now it's a time for questions. So any questions, comment, if you have.
You mentioned the expansions, John, and then some of the numbers of employees and the searches that potential companies may be doing ahead of time. On the life science side, and maybe the executive business a little bit too, where do you see some of the growth that we've had, in NC State and BTEC, where do you see some of the opportunities or things that we maybe need to be really considering, making sure that we do have the workforce base that we need? I work with the North Carolina Biosciences Organization. We've done some studies with our member companies, getting some feedback from them on what they anticipate there needs to be, and that's going to be a big push for us. So just wondering how you see that playing out.
Sure. You know, economic development used to be much more straightforward. It was find a patch of land, some green grass. And from the state perspective, even 20 years ago, what we would do is really provide water and sewer, kind of the basics, basic utilities to that property, and then the company comes in and builds it on their own, and they go out and find the employees on it. All that has changed. Today, we are having to be much more engaged. We're having to, it's a data-driven world like it has never been before. And so it's really on us to show them not only what the workforce looks like now, but where's it going to go in the future. And so for us, in any of those opportunities, it is critical to have the community college at the table, for example, to talk about the customized training programs that they offer for these businesses, because this is something that we know not every other state works with. And it is amazingly powerful for us to have a letter of support from the community college in each of the proposals that we put out. That's something that we know other states cannot do. It is getting more and more complicated, though. It's not just enough to provide information about the workforce and the basics, the connectivity that you're going to have at this location. We're having to get into areas that I don't think economic developers ever thought that they'd have to discuss, like housing, like education, like healthcare. I mean, these are all things that businesses want to know. Businesses are, our clients, and due to the fact that they have access to a lot more data than they ever did, they can tell what the scores of schools in any county in North Carolina are. They can tell what the healthcare ratings are of the facilities, and they are going to go where they're going to be able to find the best employees, but they're making an investment, and they need to make sure that they have everything that they need. So those are part of the challenges that we deal with. But it's also really important for us to be able to show the interaction between industry and and academia as well. And so here, we're blessed with the relationships. I mean, anytime we've got a new business coming in, we're typically working with one of our major universities to bring them to the table to talk about the research that they're doing, to talk about how they can engage with this business, to talk about the programs that they're working on. We brought businesses over to NC State Centennial Campus to show them the Non-Wovens Institute. A lot of folks will walk up and say, John, this is the Non-Wovens Institute. Why are you taking me to a textile plant here on the campus? And I said, no, you've got to see what's going on inside here. You walk in and see what's going on with antimicrobial materials and medical devices and recycled materials and things like that, and it's absolutely amazing. And this is something that it has a terrific impression on information technology companies, biotech, and many, many others. And so all of this is part of what we've got to be prepared to do now, and there'll be even more in the future.
Any other questions?
wanted to ask another question for John specifically, and anyone else who wants to chime in. What can we be doing better as a state to be able to more efficiently partner with overseas markets, specifically Japan, of course, but also other countries? Honestly, I don't want to say this, but where are we lacking where we need to really try to bridge the gap between competitor states? What can we be doing better there?
Sure. I would say the challenge for everybody in the room is to ensure that North Carolina is the most globally engaged state in the Union. That's a very easy way for me to say it. It's an effort that a variety of folks from academia, industry, and government have been working on for a number of years now. There's a number of different ways that we do it. I think North Carolina has been one of the more progressive states in terms of our global outreach. For example, we've had an international trade office in Hong Kong for over 25 years. We've got an international trade and investment office in Tokyo for over 20 years. These are really important assets to have because, as you all know, business leaders and academics in the room, you know as you go overseas, you're trying to establish a relationship with folks. We do the same thing when it comes to all the things that we do on the government side, the economic development side. It's critical that we have those assets. What I would say is one of the major challenges for us, and we've heard it, we'll hear it throughout presentations, anytime there's a group of state agencies in the room talking about some of the things they do, or the universities, we have a lot of best kept secrets. And not enough folks do know about the things that we do. Many of you may not have known that the state of North Carolina had that office in Hong Kong for the last 25 years. And so it's important that we all learn what assets are there. It's important that people like me market the fact that we have those assets and we want people to take advantage of. Incidentally, we also have offices in Mexico City, Toronto, Brussels, Dubai, Singapore this year, South Korea, and India, just a number of fantastic international assets that are helping make your lives, the academics and the business people in the room, making your lives a little bit easier. I took a call from somebody this week who was being told that a joint venture he was looking at doing in India was going to require a certain amount of investment and working directly with the government. This person in Raleigh doesn't know anything about setting up a joint venture with the government of India, so he called me to make sure, is this legitimate? And fortunately, we have an asset in place in India who can answer that question for him. And so I'm happy that he knew about it, but not enough times do I hear. I usually hear, I really wish I had known about that earlier. And that's what we're trying to change.
Thank you.
So in the near future, why don't we have this kind of round table and workshop in Nagoya?
Yes.
And we are very open to invite many researchers and industry people from over Japan, so whole Japan. So we can be a very interface.
Yes. So this is the 10th anniversary event. So 11th event will be in Japan.
Next year.
Yeah, thank you so much. Please give us a panelist a big applause. Thank you so much.
This concludes all our presentation for today's roundtable. Thank you again to each of our technology presenters. Also thank you all the panelists for the engaging discussions. At this time, we would like to invite Dr. Steve Rommel to offer closing remarks for the 10th anniversary Annual Tech Roundtable 2019. Dr. Rommel is Associate Dean of College of Agriculture and Life Sciences at NC State. Ladies and gentlemen, please welcome Dr. Rommel.
Thank you. First of all, let me thank Nagoya University for putting this on today. I think it was a great event. I think they invited a great cross session of people today.
large companies, small companies, university folks, government folks, people in our startup community, intellectual property. I think it's a really good group of people. Hopefully we'll have some great discussions over lunch. Also, I want to thank Nagoya University for their commitment to NC State, first of all, but also the other institutions here in RTP, and to RTP with their persistence and having a permanent presence in RTP. I think that's really a validation of RTP, actually, that Nagoya University is so committed to having us here. So I want to thank them for that. I also really like the format of the presentations today. I thought, or the format of the roundtable today, I thought the presentations were fantastic, and I really think it's a great venue to stimulate some thinking, some science, some collaborations. I will admit, these presentations made me a little melancholy. I'm an administrator now, but for 30 years I was a researcher. And I'll tell you, the talks I heard on light sheet technology today and on the cyclic peptide library work, and I would be all over these folks if I still had a research program collaborating being a biologist. And also, clearly I'd also be using CRISPR technology too, maybe not so much in the applied arena, but using it as a tool to do some of my hypothesis research. So I thought the science was fantastic. So I'm supposed to synthesize what happened today and give a perspective for us when we leave. And really there's two words that I'll discuss for the next few minutes. And one is collaborations. Clearly this is what this is all about. And the other one is economic development. As the research officer for my college and being in a land-grant university, our mission has always been delivery. Do great science, but deliver. And I saw that arc today of basic science and then also delivering. Mostly delivering in the form of intellectual property and startup companies, but that's a great form of delivery because it also is economic development too. So what I saw today was really what the core ethos is of a land-grant institution, but I'm also certain it's the core ethos of many universities now. And always has been. It's just not blue sky science. It's about doing something that's relevant to your state and relevant to society. So I really saw that today. Also, I really saw a lot of interdisciplinarity, a lot of collaborations in the talks. If you look at the talks today, every presentation had collaborations, either intra-university, inter-university, collaborations with companies, collaborations with venture capitalists, collaborations with government entities or philanthropic organizations. And that's really what we're all about. There was mention of some of the grand challenges today and some of the opportunities for nutrition and things like this. To really attack these grand challenges, we have to be a lot more interdisciplinary than we are. We really have to reach out to the engineers, the mathematicians. We hear a lot about artificial intelligence and machine learning. We really have to capture all these technologies and integrate them into our biological problems and opportunities to really solve these grand challenges. And that's sort of a challenge. I don't think are doing that as well as we can. I think we really need to accelerate thinking and interacting with other organizations and entities to enhance that. So again, we can accelerate discoveries and solutions that are truly interdisciplinary. We at NC State are taking baby steps to do that kind of stuff, as is every institution, I'm sure. There was mention today of these cluster hire program at NC State University, which is hiring a group of faculty around a theme, not a disciplinary theme, but a challenge theme, and then purposely hire people in broad disciplines in different colleges to come together to look at that grand solution. I think that's one example. I think another example and challenge for us today is to increase our collaborations with Nagoya University and also with folks here at RTP is to train a different type of student. I think we did a survey not too long ago asking companies and government agencies what they want to see in a graduate. They want to see a lifelong learner. They want to see somebody who really has great depth in their discipline, naturally, that's what it's always been about. They want to see somebody who's had an international experience, maybe not necessarily speak a foreign language, that'd be great, but somebody who's been to other countries, has experienced it, understand about traveling and something outside of the United States. And then they're also interested companies by taking on a graduate program that touches all this. And to do that, then, you'd have to have an international experience, a collaboration with an international entity. You'd have to have a collaboration with a company or a startup or a major or an internship and these kind of things. And also, maybe the research project would have to be between two different investigators from two different disciplines. And that's one way to do something virtuous, train a graduate student, and create a new kind of graduate student, and to develop these interactions both internationally and with our private partners. So I'll close there. I just want to say there was a great event today, and I guess the next one's going to be in Japan, is that right? But I'm glad these things happen annually, and I think it was a great event. Again, I think there was some really fantastic science today, and I do want to thank everybody, and that's my comments. Thank you.
Thank you, Dr. Romeo. This concludes all our agenda today. Before breaking for refreshment, on behalf of this roundtable, Nagoya University would like to once again thank our sponsors, Jenkins Wilson Taylor and Hunt as gold sponsor, and BASF, Meyer Bigel, and Okaya USA Inc. as silver sponsor. Nagoya University also would like to acknowledge each of our partners, NC State University, UNC Chapel Hill, JST, NCBC, and EDPMC. Thank you all for your help in making today's event possible. And most importantly, Nagoya University and our partners would like to thank you, our audience, for being with us today at our 10th anniversary event. We hope today's roundtable and the contacts you make here will be seized for investment collaboration in the future. Thank you once again. And now let's enjoy refreshments that have been prepared for us. Thank you.